I'm very pleased to introduce Michael Berry. He was faculty with the department,
Princeton Neuroscience Institute.
Princeton Neuroscience. They have a big building there and a really nice group of researchers. But his background is primarily in physics, from originally from Berkeley and Harvard, with a little bit of philosophy and chemistry mixed in. But you'll see that he's really trying to integrate. You know, we're really good physicists and also neuroscientists. We're good at taking things apart. You know, the constituents, the molecules, trying to understand how they interact with each other. But it's much more difficult to try and put them together, right? So you have these great textbooks, thousands of pages of, for example, the connections between neurons throughout the brain. And the question is, can you try to understand the framework, the conceptual framework underlying all of that communication that's going on? And so that's a question that Michael took up first in the retina, focusing on how information is coded in the retina, and how information can be used to predict the future. Input coming in.
And what's the true data rate? What is the true data rate? Information data rate.
Yes.
Yeah.
Well, maybe if you ask that question, okay. So, Michael, take it away.
Okay. Thanks, Terry. So that was a good introduction because so I mean, my topic here is neocortical motor control, and as Terry mentioned, in recent years I've been working on what I call large-scale brain theories. So that's this more synthetic approach of trying to put together a lot of the different areas of the brain. And so this is going to be a story that really involves global brain dynamics that's distributed over many parts of the brain, like the definitely the majority of the volume of the brain. Okay, so so kind of the underlying approach I'm taking here is this idea of repeated microcircuits. So for instance, if you look in the neocortex, there's an echo here. Is that?
You need to mute your own computer. Not mute it. Turn off your own speaker.
I turned my speaker off, but I guess I can.
Yeah, sometimes Zoom take control and you have to do it in Zoom.
Let's see.
I have the same problem.
Okay, okay, good. I turned the volume down to 0. Okay. So a very good example of this idea of repeated microcircuits comes from the neocortex. Here you have layers of cells and kind of organized into columns. And so the general idea is, you look at different brain areas, you see, cell types with specific connectivity among the cell types. But if you look in kind of the larger volume of the brain region, these kind of circuits are to pretty good approximation, kind of repeated over and over. Okay, so I mean, just to be clear, this is biology, so these microcircuits are not exactly repeated. So if you look at different areas of the cortex, there are certain differences one can identify. But sort of the approach I'm taking is, let's ignore those differences for now, and I think that's potentially a useful approximation for this kind of stage we're at in neuroscience. And maybe when we understand things much better, we can look at some of the more subtle differences and gain insight from that. But I would say the big driving idea is if we assume repeated microcircuits, then if we have a detailed and good understanding of a relatively small number of microcircuits, we might gain insight about lots of what the brain is doing. Okay, so how many microcircuits are we talking about? I think this. So neuroanatomists, neuroscientists divide the brain into 7 major brain divisions, and I like conveying this to let's say non-neuroscientists, because I think, most people have the impression that the brain is just this organ of bewildering complexity, and there's just always another nucleus you've never heard of, doing something strange. Even for professional neuroscientists, there's always another nucleus you've never heard of. But this.
Collectorless. Where's the
Princeton.
Oh, I see. Okay.
Okay, so this is like a tiny bit of a cheat, but let me just mention. So among the 7 major brain divisions, the idea is that if you look at the types of neurons and the local connectivity, they're really absolutely different. Okay, so I would say at a very minimum, we need to have different microcircuit models for every major brain division. Okay, maybe we can get away with 1 microcircuit for a whole brain division, maybe, but at least 1 per brain division. And then I just for full disclosure, I put a little asterisk next to brainstem. So that is a brain division that does not have repeated microcircuits. Okay, so it has of order, maybe 100 different nuclei that all have kind of specific function. So maybe it's a little bit of a cheat hiding a lot of the complexity of your brain in this 1 brain division. But on the positive side, the other 6 major brain divisions have highly repeated microcircuits. Okay.
So Michael, you're leaving out the hypothalamus.
That's part of brainstem.
Hypothalamus, at least in your conceptual model.
In this breakdown.
Okay.
Well, the star means everything else.
Yeah. So there's a lot of there's a lot of detail left out in brainstem, but again, the positive story is these other 6 brain divisions. The vast majority of the volume of the human brain have these fairly repeated microcircuits. Okay. So and again, the story about neocortical motor control spills over most of these major brain divisions, and so just at this high level view, what I've done is I've come up with a catchphrase to describe the computation in each of these major brain divisions. Okay, so we'll start with the neocortex. Okay, it actually gets 2 catchphrases. The first one is engine of unsupervised learning. Second one, generator of motor plans. Okay, so the unsupervised learning is in gray because I'm not going to talk about it here. So this talk will be about the motor plan part. Okay, so let's dive in. So the motor planning really originates from a key cell type in the neocortex. This is shown schematically here. The cell type has multiple different names in the literature. It's in layer 5, but it's in the bottom half of layer 5, so it's often called layer 5B. Another name for it is layer 5 PT, or pyramidal tract. Okay, and that's because it sends a huge axon. That is the pyramidal tract. It's also called tufted because if you look at it, the apical dendrite goes all the way up into layer 1 and has a gigantic apical tuft in layer 1. Okay, and also this neuron has like the biggest cell body of all the neurons in the neocortex. It's really, this cell type has gotten a lot of attention in the literature. Okay, so I'm going to interpret these neurons as encoding a motor plan. Okay, and this little star here in the top means this is a hypothesis or interpretation I'm making. Things without a star are like truth.
But motor command or motor plan?
Motor command. It can be both. Well, you'll see. Okay, so let me just try to give a little bit of evidence to reinforce that, because I think a lot of people don't have the intuition that neurons in, let's say, the visual cortex are encoding motor commands. And this is really very important for the talk. So I'll cite a couple pieces of evidence. One piece of evidence comes from a series of studies from Peter Schiller, where they did microstimulation in V1. And to kind of summarize a few papers, if you stimulate in the superficial layers, layer 2, 3, it causes distraction from visual targets, and they make an argument that it's similar to kind of causing visual phosphenes in humans.
Distraction from visual targets?
Yeah, if the animal is doing a task and there's a target in the receptive field, if you microstimulate in layer 2, 3, the animal doesn't do the task.
And
It's like it
like
doesn't
on
notice
other layers. If you stimulate in layer 4, for instance, it thinks there's something there, right? It's very
Yeah, so
yeah.
so the deep layers, if you microstimulate there, you get saccadic mind movements. Okay. So for instance, there are these layer 5B neurons in primary visual cortex, where they project is to the superior colliculus. Okay. So that's the motor output pathway of primary visual cortex. Okay, so kind of some other evidence. There are a couple nice papers from Tony Zader, where he was looking at the projections from the primary auditory cortex to the striatum.
What's the striatum?
It's the input to the basal ganglia. And so he had rodents doing a simple task where they listen to an auditory cue, and then they lick left or lick right, depending on what the auditory stimuli was. And what they showed with, they specifically labeled layer 5 neurons that project them. It biased the choice of the animal. Okay, so that's saying that these neurons in the animal doing the task are actually driving, in part or in whole, driving the behavior. They also showed that as the animal learned the task, the synapse from the layer 5 neuron to the striatum potentiated, and that was related to how well the animal did the task. Okay, so this is evidence here that these neurons in primary auditory cortex are projecting to brainstem nuclei that are making movements like lick left or lick right. Okay, there's other evidence, but I just wanted to include a little bit of that. Okay, so now one of the really important things about these layer 5B neurons is that they're present in all cortical areas. Okay, so I like to think of this as maybe the most important fact about the brain that many neuroscientists don't seem to know. I don't exactly know what all of the neuroscientists know, but I feel like when I say this to people, a large fraction of the time, neuroscientists are like, really? Okay, it's super well documented in the literature. So I'm not sure why most neuroscientists don't seem to know this or incorporate this into their ideas about the brain. But one suggestion is we have these names like visual cortex and motor cortex. Like once you name this part of the cortex visual cortex, you just sort of implicitly think it's doing visual processing and not motor, especially because there's another area called motor cortex. Okay. But these neurons and these pathways are present in every cortical area, primary sensory all the way up to prefrontal. Okay, but that leads to a massive problem. If all these different pathways are potentially generating movements, you have this problem of if you're to do them all at once, it would be a disaster because it'd be all kinds of incompatible movements. Maybe it'd be like an epileptic seizure or something. So you have a big problem of coordinating all these different motor outputs from different areas of the cortex. Okay, so the solution I'm going to argue is a balance. Yeah, please.
Yes. All of layer 5 cells also project into the caudate.
Yeah.
And they are mapped roughly in line with the areas of the cortex. So the input to the caudate, any local area of the caudate comes from a specific region of the cortex. Are these the same cells that are projecting to the spinal cord, or is this a dual?
There are two major cell types in layer 5.
Right.
So there's another cell type that also projects to the striatum, but these layer 5B cells project to the striatum.
Yeah, following the map.
Yeah, with a precise
and they also project to the spinal cord.
Yes.
So it's a collateral going into Okay, that's what I wanted to clarify.
Exactly, yeah.
Are they the same cells? They are.
Yeah.
And they're just sending collaterals.
Yeah, I mean, there's there's a whole bunch of evidence in the literature that that's the case. You know, people fill cells, they see the collaterals going both places. You know, there are at least some claims that there might be some different cell types that don't send collaterals everywhere, but the majority of the literature makes it look like these guys send collaterals to all the different targets.
Mike, can I make a survey? How many people know what the caudate is? Anybody know what the caudate?
Okay, it's
Yeah.
it is a part of the striatum. So
You can ask the same thing. What is this striatum?
okay, striatum is the input to the basal ganglia, and I will go over all this. Okay, I will actually. Okay, right. So in broad terms, I'm going to say the solution to this problem of incompatible movements is through the thalamus. So these neurons, they send axon collateral into the thalamus. So the thalamus is getting an exact copy of this motor command. Okay, and then related to what Barry was just saying, these neurons also have projections to striatum and cerebellum. Boom. Okay, so these very same neurons project to these kind of three different major targets in the brain. These are three different major brain divisions. And all of those major brain divisions send excitatory input back to the neocortex, okay? So there are three major recurrent excitatory loops that are the basis of a lot of motor control, okay? Okay, so go to the thalamus. So, you know, it's this kind of interior structure underneath the cortex. It's bilateral, looks a little bit like a football. So the catchphrase is communication hub of the brain. Okay, so now the thalamus is, so the thalamus in the literature has many different nuclei, okay? But what I'm looking for here is kind of an organizational scheme to help understand the different nuclei. So the idea is there are four major divisions, kind of core relay cells, matrix, intralaminar, and then around the thalamus is a sheet of reticular nucleus. So maybe you don't really want to count that as another division. And there have been kind of two major approaches to categorizing all the different parts of the thalamus, Murray Sherman and Ted Jones, and so I'll go over those quickly. So in Murray Sherman's categorization, there are two types of nuclei, first order and higher order. First order nuclei get subcortical driving input. So an example is the LGN that gets input from the retina, or MGN, which gets input from the auditory brainstem. The other kind is a higher order nucleus. In that case, the driving input comes from the neocortex itself. Okay. Examples are the pulvinar, medial dorsal nucleus, etc.
I thought that every area also got input from cortex too, like the LGN gets feedback from the cortex.
Yeah, so this is with respect to what's called the driving input. And I'll talk about that in a couple slides later.
Positive.
Okay, sorry, what?
Positive. Excitatory input. Or driving is the same as excitatory?
Let's get to it in a couple slides. Okay, so then in the Ted Jones version, what he found is that if you label different thalamic relay cells, they express different molecular markers. It's very clear there are a few different cell types. One he calls core. So those neurons in the thalamus, they project to intermediate layers of cortex, layer 4, which is like the sensory input layer. And then matrix is another type, and it projects primarily to layer 1 and also a little bit to some other layers. Okay, so this is going to have different effects on the cortex and the recurrent loops because of these different projections. Okay, so and then I guess there's more recent work that I think does a little bit better categorization. Maybe there are 4 types of thalamic relay cells: core, like we're talking about, So here's kind of a nice picture. It goes up and it has kind of a dense focal projection, most of them are 4. There are 2 types of matrix cells. One of them has a fairly focal projection in layer 3 and also layer 1, and then multi-areal goes to layer 5 and also has connections to other cortical areas. And then intralaminar, we're not going to talk about. Okay, so the core type is relaying sensory information into the cortex. And this is like the LGN, but this is actually fairly uncommon. And most of the thalamus is.
Is there a reason for that name?
Matrix?
Yeah. Is it supposed to represent that it's some matrix vector multiplication, or that it's arranged, that it's arranged in some periodic way? Like it's illustrated there with that repeated connectivity?
I think in some of his early staining work, he saw like little blobs that were core, and the rest of it spatially was matrix. But I don't think I think the name core is kind of useful and matrix is kind of confusing. So sorry about that.
I have a slide, but if people want to ask me, I can show you. That illustrates where the names core and matrix came from.
Okay, good.
And it had to do with what the histology, the staining the tissue looked like.
Yeah.
It's just a description of what it looked like to the person who was doing it.
Yeah, I mean, for non-neuroscientists, I have to apologize for kind of nomenclature in anatomy because it's really a mess. Okay, so now getting to Toby's question. Okay, if we consider these relay cells in the thalamus, you can distinguish 2 major types of excitatory inputs: drivers and modulators. So in the LGN, Murray Sherman first figured this out in the LGN. So the retinal ganglion cells, they constitute only 5% of all the synapses onto a thalamic relay cell. But when you measure receptive fields, the LGN cell is like nearly identical to the retinal ganglion cell. And so what's happening here is a single retinal axon branches into like 100 boutons that are all making synapses onto the soma of the relay cell. So it's basically like a neuromuscular junction, completely deterministic. So the other input, it accounts for about 50% of all the synapses onto the relay cell. This one is coming from layer 6 of the primary visual cortex. But the issue there is that a given LGN relay cell will get this input from, get maybe 1 or 2 boutons from a single neuron in the visual cortex. The inputs are far out in the dendrite. And in addition, they have mostly metabotropic glutamate conductance instead of ionotropic, so they cause a small EPSP. So even though there are lots of these synapses, they end up having a very weak influence on a given relay cell. Okay, and Sherman has done some nice work, measuring properties of synaptic transfers and slice from many synapses, and they fall into these 2 clusters: the driver and the modulator. So the main things that distinguish them is how big is the EPSP. Drivers have big EPSPs, modulators are small. Depression, the drivers depress. Metabotropic glutamate, the modulators have it, the drivers don't. Okay, but so the main issue is, the drivers are what are really going to cause the spikes. Okay. And here's just another picture of cortical inputs into the thalamus. This is kind of a driver synapse. It comes from layer 5B. The modulators come from layer 6. Okay. Okay, so that's description of kind of anatomy of the thalamus. So now, what is the local computation? So the idea is that the driver inputs they're very strong. They fire spikes pretty deterministically. For retina LGN, there's no report of synaptic plasticity. I'm not sure if for some other Maybe in other thalamic nuclei there are, but with really strong inputs, it doesn't necessarily make sense to have plasticity. Modulators are weak. They kind of slightly boost the firing of driven cells. They have strong synaptic plasticity. Okay, so the basic computation in the thalamus is lateral inhibition. Okay, so if you look at the excitatory neurons of the thalamus, these relay cells, they make no collaterals within the thalamus. Okay, so it's the local computation. It's a single feed forward pass. It's probably like the simplest local circuit computation almost anywhere in the brain. It's just a little bit of lateral inhibition. So the source of that, there's some local inhibitory interneurons, but in addition, there's that nucleus reticularis. Okay, so all of the relay cells send the collateral to nucleus reticularis, which then sends inhibition onto other relay cells. Okay, so lateral inhibition. Yeah, so for instance, in LGN, roughly half the retinal spikes cause an LGN spike because the lateral inhibition is canceling the other half of the spikes.
So that's weird. That's only half, If it's so important to transmit this precious visual information under low light conditions, for instance, why lose half of them? I mean, you're introducing a tremendous amount of noise at that stage.
Yeah, I mean, so the thing is the retinal population has high redundancy, so it's fine.
That's true.
Okay, so then, like, what is the thalamus doing? It has this super simple, boring feed forward computation. So the main thing it's doing is really comprised in the feedback. So let's talk about the feedback. Right. Yeah. Okay. So here we're going to mostly consider these matrix type relay cells that are multi-areal. Okay, so the output goes to layer 1 and also to layer 5 of cortex, forms recurrent excitatory loops. Okay, now here's kind of the key assumption. I mean, it's known that these synapses from thalamus back to cortex have high synaptic plasticity. Okay, so here's the idea. Let's consider a specific subset of the layer 5B neurons that encode one specific movement. And I'm going to denote that as layer 5B star. That's a subset for particular movement. Okay, so the idea is, those neurons, they feed into the thalamus. They're going to deterministically fire a subset of neurons in the thalamus. And I'm going to call those thal star. Okay. So now the idea is for those particular thalamic neurons that feed back to cortex, they have plasticity that potentiates their synapses onto the same layer 5B neurons that cause them to fire. Okay, and the reason for that is heavy plasticity. They're firing together. Okay, so the idea is this feedback is through plasticity over time. It's going to kind of strongly emphasize excitation back onto the specific subset of neurons that drove them. So the consequence of that is that it forms potentially an amplifier circuit, okay, because it's specifically re-exciting its input cells.
So Michael, I don't know at what level of abstraction this is, but as far as I know in the visual system, layer 5B cells do not contact cells in the lateral geniculate nucleus, and the feedback loop is from layer 6 cells down to the thalamus and back up to the cortex. So you lumping layer 5B cells with layer 6 cells because the 5B cells project to the superior colliculus.
Okay, so remember there are these different thalamic nuclei. So the LGN is a first order nucleus that does not get driving cortical input.
Yeah.
Okay, pulvinar does. So these layer 5B cells do send driving input to the pulvinar.
Okay. So this is the matrix you were saying.
Yeah, so this is thalamic matrix cells or higher order thalamic nuclei are the ones that are getting the cortical input.
Oh, okay.
So the LGN, MGN, VPL, VPM, and posterior ventral lateral, those are the only parts of the thalamus that are core type. So again, like LGN is the best known thalamic nucleus, but it's like a very misleading example for what I think most of the thalamus is doing. And then and remember these layer 5B cells, they send collaterals to these, both the superior colliculus and let's say the pulmonary, for instance, visual cortex. Yeah.
I think if I understand, so but correct, like the cortical feedback to thalamus is this weak modulatory input?
No.
Sorry,
There are two feedbacks. There's a driver that's strong from layer 5B. That's what I'm mostly talking about here. There's a layer 6 that's weak that I'm going to ignore for the rest of the talk.
I see. And so this is the strong driving input that can make the neurons in thalamus fire,
Yes.
which
Which then feeds back, fairly deterministically. So then the idea is you get this plasticity where they're going to strengthen their synapses onto the cells that drove them.
They're gonna wire themselves together, basically, right?
Into a loop. So right. So the picture is you have these kind of individual thalamocortical loops that each encode a specific movement. Okay, and a given brain area will have like multiple of these loops. Okay, so now competition among the loops. So the basic idea is that there's a winner-take-all dynamic. Okay, so here's loop number 1. It has recurrent excitation with the thalamus. Loop number 2 also does, but then those thalamic relay cells have lateral inhibition. Okay, so these two loops are going to compete via lateral inhibition in the thalamus. Okay, and so the idea is that whichever thalamocortical loop has the most activity is going to drive down the activity of the other loops, which in turn disinhibits the first loop, the strong loop, and so the loop with the most activity wins this dynamic competition. And then the idea is the winning loop triggers the movement that it encodes and suppresses the other loops. Okay, so the idea is through this winner-take-all dynamic, you get this global motor coordination. Okay, so all these different movements can't occur at once because they're all mutually inhibiting each other. And when one of them starts to get strong, it knocks all the other ones down. So it's only one loop can win the competition.
But it supposed to be local competition, right? Because you don't have inhibitory cells going long distances in the thalamus. Right?
Yeah, it's pretty local in the thalamus, but the thing I didn't mention is that these relay cells, when they project a cortex, they actually project broadly. So they're these multi-areal, multiple cortical areas. So through maybe a couple jumps of the loop, you can communicate across the whole neocortex.
What kind of movement are you talking about here? And specifically, like a reaching movement,
Yeah.
eye movement?
Click left, click right, eye movement to there, eye movement to there, global walk over here, and any of those things, all movement. Oh, okay, what? Important distinction, I'm talking about voluntary movements here. Yeah, this is all voluntary movement, yeah.
What happens if you just excite that 5 neurons without any task? So the animal it's not trained on any task.
Yeah, so I mentioned in primary visual cortex, if you do that, you make a saccadic eye movement.
Oh, okay.
Okay. With no task.
Oh, it was without task. Okay.
Yeah, no task. And so in general, the idea is that different specific cortical areas will have their layer 5B neurons wired up to different movement, different parts of the brainstem or spinal cord. Okay, so the effect you get of a microstimulation, that is dependent on which cortical area.
Okay.
But there's this overall theme that they all have layer 5B that are potentially making movements.
But is it possible that rather than encoding for movement, cell 5 is more like the output of that kind of computational block? So let's say that a computational block that more general behavior is to encode somehow, predict some kind of attention or something like that. And therefore changing L5, so it's like driving in the context of maybe one of the tasks, attention to that particular thing and moving. No.
I guess in principle, I guess, Why don't I kind of go through the whole story, and I can kind of circle back to like where I think there are some other kind of possibilities, but the idea for now is like, these neurons, they send an axon to a movement area. So, once they fire strongly, they'll make the movement. And, when their activity is lower, they're not making the movement. Maybe you want to interpret it as like a motor plan at that point. Okay, so I've started to put together a simple computational model with some of these features, but I want to talk a little bit more about the basal ganglia before diving into that model. Okay, so basal ganglia, another major brain division. Its two main functions: gating of voluntary movement, which we'll talk about, also origin of reward learning, which I'm not going to talk about. Here's kind of a simplified circuit of the basal ganglia. Got these layer 5B neurons that go to the brainstem. They send a collateral into the striatum. This is the input area of the basal ganglia. The striatum has inhibitory neurons that project to the second area. This is actually the output of the basal ganglia, the pallidum. Inhibit the pallidum. And the pallidum has inhibitory neurons that inhibit the brainstem. Okay. So to wrap this, we're going to start with the pallidum. The pallidum has, a very special type of neuron that fires spontaneously at pretty high rates, at 10 to 40 hertz. And they have these like fabulous mechanisms to decorrelate from each other. So they're all decorrelated. And they're inhibitory. So what that does is it causes a tonic or active inhibition of movement. Okay. So the idea is, as long as the pallidum is active, it doesn't matter what these excitatory commands are to the brainstem. It's going to be blocked by the inhibition.
And why is it important they're decorrelating themselves for this function?
Okay, don't worry about it.
Okay.
Let's get that. Okay, so the basic idea is you can't make a voluntary movement without pausing activity in the pallidum. Okay. How do you do that? Well, with the striatum. So in the striatum, you have neurons have very transient, sparse firing. So the idea is, you get a little burst of firing in the striatum. It will inhibit or pause the population in the pallidum, and then that will release the movement. Okay. So the idea is, it's not enough to just send the excitatory command to the brainstem. These layer 5B neurons have to also fire the striatum. Okay, so why have this weird kind of Rube Goldberg of a version of a system where you have this double inhibitory pathway, blah, blah, blah. The basic idea is to suppress or prevent unwanted movements. Okay, so the idea is, if you just had like an excitatory pathway that had to exceed a high threshold, like sometimes there'll be spontaneous fluctuations and you'll get the movement when you don't want it. So the idea is you have this massive inhibition that is totally shutting down movement unless very specific circumstances are met in the striatum. Next question.
Yeah, so this in this neurodegenerative diseases that cause involuntary movement, is there like a relation between some function between this sub-area?
Yeah. Okay. So something I haven't included in this talk is how about if I circle back to that, because okay, so let's look at the striatum. So it has a principal cell type called a medium spiny neuron. About 95% of all neurons in the striatum, they're inhibitory projection neurons. Okay, but the interesting thing is they have a high density of a special ion channel called an inward rectifier potassium channel. Okay, so this is one of the only cell types in the brain that has this channel. And what does this do? It creates a down state. Okay, so these are potassium channels, and they're gated or they open when the membrane goes more negative. Okay, so basically what happens is if you go a little more negative, you open this channel. It's potassium conductance, so the current that flows through it causes voltage go even more negative. Okay, so it's actually a positive feedback mechanism that brings you down to a super low membrane voltage. Okay, it's called a down state, or you could almost think of it as like an upside down action potential. And so the consequence is that these neurons have a very high threshold to fire. Okay, because of the sine channel. And just a little bit of data, there are a lot of physiology experiments. You inject current into these neurons, and Rio-base is the level of constant current injection to get the first spike out of the neuron. And in the layer 5B pyramidal cell, maybe 30 to 100 picoamps. Medium spiny neurons, almost 10 times bigger. Okay, so what's the consequence of that? Well, it allows for an interesting regime. So let's say the layer 5B neurons are firing at kind of a moderate level of activity. Then one thing that will happen is you won't get a movement, okay? Because it's not high enough activity to get out of the down state of these neurons in the striatum. Okay, so there's no movement, but the spikes go to all the other targets, okay? So other downstream targets are all activated as if the movement was occurring. And so the basic idea is that this is a regime that's kind of like simulation of the movement without doing it. So that's the idea of like why you have this mechanism in the striatum. It allows for this regime where you're activating almost all of your brain circuits like they would normally be activated for movement, but you're not moving.
Are you saying that if I think about closing my finger like this, then I'm doing that? That's activated?
Yeah, probably.
Okay.
Okay, so why simulate? Couple ideas. One, this generation of sensory evidence in favor of that movement, and this is going to connect to this model of Neocortical loops I'm going to show you in just a moment. Another idea is that you can estimate what would happen if you did the movement and determine if that predicted consequence matches what you want to do, and then boost the motor plan if it does match your goal. And so that's a role for the cerebellum. We'll talk about later. For time. I think I'm actually not going to get to this. That's fine. Okay, so all right. Another very important feature of basal ganglia is that there's kind of one other branch here that I left out, which is that the pallidum also tonically inhibits other regions of the thalamus. Okay. And so if you activate striatum sufficiently to pause the pallidum to make the movement, what you'll also do is you'll disinhibit an area of the thalamus, okay? And the thalamic relay cells have a low threshold calcium current that causes them to fire a big burst of spikes when you relieve them from inhibition. So then the thalamus will have a very strong firing that feeds back to cortex when you have a movement. Okay. So why do that? So the idea is that this feedback is going to communicate to the rest of the brain that you're doing this given movement. Okay. And why is that important? If every area of the neocortex is making motor plans constantly, if you actually decided to do a certain movement, you're going to have to change your plans or modify them based on the fact that you've actually moved your body over to here. So the idea is like all of the cortex needs to know that you have decided to do this movement. Okay, and what's interesting is there's some fairly recent evidence where people are seeing this phenomenon in the cortex, and it's kind of the field is finally seeing it now that we have some more wide field kind of recording techniques for cortex. This is one example of a really nice paper from Ann Churchland's lab. So what they're doing here is they have a mouse. It's doing a visual task where it like licks left or licks right. But what they're doing is they have a bunch of video cameras. Okay, so they're videotaping the face from different angles, and they're looking at the paws and measuring running on a ball. And so they identify movement variables like, whisker twitching or, or scratching or, like all these things that are unrelated to the task that the mouse has to do. And when they have all these movement variables, then they just do a big general GLM, linear model, to kind of regress on all those other movement variables and see, how much of the activity of neurons is explained not by the visual stimulus and the visual task the animal's doing, but based on all these unrelated movements that are unlocked to the task. Okay, so what they find is, first of all, when they trigger on these sort of movement events, they see activity all over the cortex. Okay, and then another thing they see is that activity that they measure is better explained by these unlocked movements than it is by the visual stimulus, by at least a factor of 2, even in V1. Okay, so basically, and there are other studies like this that suggest that the majority of activity in the cortex is actually encoding these movements. It's like, most of this.
To remind everybody, you heard a lecture yesterday. Lisa, is she here?
Yeah, but she's.
Oh, okay, hi, okay. So she also had that projection from the motor system to V1, and there was part of the control, right, to subtract self-generated sensory signals.
Yeah,
which is similar to I think what you're saying.
Yeah, I mean, well, I would say that's another potential function, or maybe it's kind of similar to what I said. Okay, but anyhow, this is what people see experimentally. It's kind of massive global broadcast across all of the cortex when the animal makes a movement. Okay, okay, so now I'm going to model a fun cortical loops, and got my clicks out of order, so let me just put them all up. Okay, so here's our layer 5B neuron with reciprocal excitation with the thalamus. Okay, so first step, we're gonna talk about like a pool of layer 5B neurons, that are the neurons encoding a specific movement, and we're gonna represent their activity with a time-dependent firing rate. Okay, so that's like the simplest thing you could imagine doing. There's actually an argument based on a much more complex argument based on structure of the population code that says this is a good way to do it. But in the interest of time, I'll skip that, and let's just say, this is a pool of neurons representing one movement. We're going to talk about its overall firing rate. Okay, so now the dynamics of kind of a generator potential. And now you have some additional inputs to the neuron. So the first input here is there's local recurrence within layer 5. So that's the strength of that is parameterized by gamma. So you get some excitation, that's your firing rate times gamma. That's an excitatory input. You also get feedback from the thalamus. That is the firing rate of thalamus times this constant alpha. And then in general, we have like some other inputs. Here, eta will just be a constant. Okay. Okay, so now the other important detail is that the input into layer 1 has been shown in a number of studies not to have that much effect on the spiking of a layer 5B neuron, but mostly to change the gain of the neuron. Okay, and so the input in layer 1, which is a descending input from other areas of cortex, we're going to model that as a gain control on the neuron. So the idea is that the firing rate is a rectified version of the generator potential times the gain, 1 plus G. Okay, it's a gain control. And so now in the thalamus, we're going to, the idea is that this input just drives the neurons in the thalamus, okay? And maybe there's some lateral inhibition from some other activity that is this variable theta. So then the firing of thalamus will just be this activity of the layer 5B neurons minus the threshold and rectified. Okay. So it ends up being mathematically a very simple model. We can rearrange terms where there's an effective gain and this other input. And so this variable omega is the effective gain. So it depends on recurrent excitation from thalamus, recurrent excitation locally, and it also depends on the gain control from cortex. Okay.
And what happened to the rectifier? Okay, in the
Oh,
bottom you have a linear equation, but that's totally nonlinear.
Sorry, this is assuming you have activity that's above these thresholds.
Okay.
So, if this has a positive activity, you'll always be above that threshold and assume theta is small. Okay, so you get some different regimes of dynamics depending on this effective gain. Okay, so if this variable omega is positive, what's happening there, I should have rewritten the formula, but in that case, the leak is bigger than the positive feedback, okay? So when the leak is stronger, you're gonna just reach of activity. Omega 0, in that case, the positive recurrent excitation exactly matches the leak. And so you're going to have linear ramping of activity. And then omega negative means the positive feedback is stronger than the leak. So you're going to have exponentially rising activity until you hit the threshold. And one kind of another sort of overall idea here is you want the cortical loop to be under control. Okay, so if the cortex is giving no input, this G factor, G variable is 0, then you want to be in this steady state regime. Okay, so the idea is when you have a cortical input, you activate the loop, kind of turn it on. But if you don't have that, then it's not always ramping to threshold. So this kind of ramping dynamics is under control from the cortical gain. So as long as this is true, you're not going to spontaneously ramp with no cortical effect. Okay, so now what happens if there are two loops? So here they are competing in the thalamus, and then whenever the activity gets high enough to trigger the threshold in the striatum, then you get the movement. And so this is just a simulation of a model where the two loops have the same intrinsic properties, okay? But the cortical gain for one loop is just a tiny bit higher than for the other loop, okay? So what happens there is, like, initially both of the loops start to ramp together, but loop number 1 has slightly higher gain. So it starts to have more activity, but then it inhibits loop 2, okay? So the difference grows and grows. And then at kind of a later point, you really shut off loop 2. And then loop 1 actually has like a steeper ramping. It's kind of won the competition. Or once it's shut off the other loop, and then it goes up until you reach threshold. Okay, so this kind of ramping phenomenon, is the type of thing you see when people make spontaneous movements. So this is related to what's called the readiness potential. So here's an example of an experiment to measure the readiness potential. You're sitting there, there's like a button, and your task is press it whenever you feel like it. Okay. So there's no external cue. So, the person is deciding to spontaneously press the button. Okay. So while they're doing that ultra simple task, if you measure EEG over their whole brain, what you see is, of order a second or more before the time at which you actually press the button. Okay. This has been known for decades, that you get this overall increase of scalp voltage ahead of a spontaneous movement. And so, okay, so the idea is in this model of thalamocortical loops, if we set their gain to have omega close to 0, and we just have this input as constant, they're going to do this sort of ramping behavior. Okay. this is, a review article looking a little bit more at kind of ramping. It's not just in the EEG, you also see ramping in firing into individual neurons. Okay, but the idea is that this ramping regime of the thalamocortical loop is qualitatively in agreement with the phenomenon of the readiness connection. Okay, so in another limit, here again, let's say we're in this integrator or ramping regime. Now, if the input is a sensory stimulus, okay, then the activity is literally the integral of the sensory stimulus. Okay. So in this regime, the thalamocortical loop that's getting the time-varying sensory input is integrating in sense of calculus the stimulus. And so, for instance, you could think about the case where there are two different categories for the stimulus, and you're trying to discriminate. So, for instance, there are all these famous experiments where monkeys are looking at kind of randomly moving dots, and a fraction of the dots are coherently moving to the left or to the right. The monkey has to look at that and decide whether the motion is to the left or to the right. And it's an example of kind of integrating sensory evidence in the neuroscience literature. So just for instance, imagine your stimulus is kind of a Gaussian variable, some mean and standard deviation, and you take the positive part of it and send it into one thalamocortical loop, and you take the flip negative part and send it into the other thalamocortical loop. Then the idea is these loops will integrate the sensory variable to see is it more positive or is it more negative, and so here's kind of an example of a simulation where we have this Gaussian variable with just a little bit of a bias, positive, and the dark green is the loop that has the positive input, and it's nearly the same because the bias is very small. But if you have a stronger bias, then the loop reacting to the positive component will ramp quick the other loop, and so that will be a decision that the motion is to the left or whatever. Okay, so the idea here is that in this sort of limit, the model is in qualitative agreement with what's called the drift diffusion model of decision making. It's kind of a model that's been applied to many different neural systems. We're kind of integrating sensory evidence up to a threshold. And so that's what the thalamocortical loop is doing in this case. Okay, let's see. So summary of thalamocortical loops. Okay, so the idea is a single thalamocortical loop is like an amplifier circuit. The actual kind of gain or degree of amplification can be controlled by a cortical input, using a cortical input. So you can get into a regime where the gain gives you positive feedback that matches the lead. Ramping of activity for a spontaneous kind of nearly constant input. And so, that's qualitatively in agreement with ramping activity you see in neurons and kind of decision-making tasks in the brain, and also the readiness potential. And then when there are multiple loops, there's this winner-take-all competition, and that implements a form of global motor coordination where only one loop can win and make the movement. Also, in this regime, you can have integration of sensory inputs and do kind of a sensory decision-making task based on which loop has the bigger interval. And so that's qualitatively in agreement with the drift diffusion model of decision-making. And then in addition, when one loop wins the competition, you get this global broadcast that spreads that information throughout the brain. And just mischievous thought we could talk about later. Some evidence this might be related to the neural basis of consciousness. I don't have time to make that argument, but I'd be glad to discuss that at a later point.
By the way, how much longer? Because you're reaching an hour.
I have one more slide and then I'm done.
Oh my God.
Okay, so I just want to show a little bit of nice experimental data about the importance of the thalamus in kind of a task. So this is data from the Svoboda lab, and they published a whole series of papers using the same task. So there's a rat or a mouse, I think. It's in the darkness, and what happens is they put down a pole next to its snout, either near or far, the red location or the blue location. Okay, and the animal whisks that, and it uses the whisker, information to decide was the pole in the red position or blue position or red position. Based on that, it licks left or licks right. Okay, so it's a somatosensory task using the whiskers to decide whether to lick in one direction or the other to get water. Okay, so right, and maybe. Okay, just a little bit of background. In some other papers, they sort of inactivated all over the cortex, and they found a particular cortical area called ALM that is necessary for the task. Okay, so let's say this is mostly being done by ALM, but then ALM has reciprocal connections with a particular part of the thalamus, and so if they use optogenetics to inhibit that thalamic nucleus, completely messes up the task. Okay, because the ALM is bilateral and it triggers a contralateral licking movement. So basically, if you inhibit the thalamus, then it never licks in that direction. Okay. And you get similar results for optogenetically inhibiting ALM itself. Okay. So both the cortical area and the thalamic nucleus have a similar impact on behavior. If you look at activity in both thalamus and cortex, you see ramping activity, before the movement. There's a lot of neuron to neuron variation, but if you kind of average over the whole population, you get pretty nice ramping signals. Okay, another interesting thing they did is they inhibited the thalamus and recorded in the cortical area that has connections. What happens when they do that is that cortical activity crashes in like 10 or 20 milliseconds. Okay, so it's saying that this recurrent excitation from the thalamus is utterly necessary to maintain cortical activity.
So, Michael, I don't think there's any evidence that it's the recurrent excitation from the thalamus. It's just excitation from the thalamus. This will happen in all cortical areas if you inhibit their specific thalamic nucleus in an awake animal. Well, this and LGN, it'll be one after the other.
Yeah, that's what I mean. So just the excitatory pathway from thalamus back to its reciprocal area of cortex. If you silence that with optogenetics, then cortical activity crashes.
Yeah,
and very fast. It's kind of amazing. And then just one last thing. They a slightly different experiment. They looked at what happens if you inhibit the thalamus. What happens in ALM? The primary motor cortex, which has a big cortical connection to ALM, and basically silencing thalamus has a massive effect. Silencing M1 does almost nothing. Okay, so the thalamic input is much more important than the input from an important neighboring cortical area. Okay, so I'm just showing some of this data. Thalamus. Basically, if you inhibit a specific area of thalamus, you just can't get the movement corresponding to that area of the cortex. Okay, and then I'm gonna stop. I'm gonna just go to a summary slide. There it is. Okay, thank you. Okay, so we do have time for a few questions.
So this lovely simple diagram of this multiple roots. What does that look like for the actual system itself? Are you imagining like a small population of neurons, of L5 neurons that don't talk to each other? That's why you would have the separated, roots. Yeah,
yeah, I mean, so the idea is that in the entire Layer population, there's a bunch of correlation between the neurons, and that causes there to be well-defined clusters of activity. And those clusters are the pool that corresponds to single movement. So the separation is actually based on kind of the statistics of the population code. It's not that they have no connections to each other.
What percentage of neurons are each of these clusters? Like 10%?
Okay, so I've done a lot of analysis of these population codes in retinal ganglion cells, and that's about it, about 10%. Roughly. Kind of a single cluster will have about 10% of the neurons with relatively high activity for that cluster, and those neurons have lower activity for other clusters.
I have two quick questions. You never mentioned the prefrontal cortex and its relation to the basal ganglia and voluntary movement, which is one of the things you have.
Yeah, I mean, so I mean, the prefrontal cortex has the same layer 5B neurons that go to, their own part of the basal ganglia, their own thalamic nucleus. In this case, it's the medial dorsal nucleus primarily. So I mean, at this level of approximation of repeated microcircuits, like the same sort of thing is happening there. Okay, but what you might be wondering about is that in the motor system, there's some degree of a hierarchy. So prefrontal cortex has a bunch of, let's say, inputs that descend and affect M1 motor cortex. In fact, this like descending cortical gain signal, like for instance, it could be coming from prefrontal cortex.
And other quick question. We had a nice talk here last week about the control of speaking, for example, and how inputs coming in from the auditory areas going into the motor areas. And I just wondered where in this loop you have, is it the same layer 5B that provides the inputs into the motor areas for articulatory control?
Well, I mean, let's say the primary auditory cortex, will probably have some direct cortical inputs that will go into M1 and vice versa. So there'll be those pathways.
So those are all unrelated to the things you mentioned, or is it?
They're not really unrelated because, if you think about, I kind of gave you this picture of like a tiny little area of cortex, a tiny little area of thalamus with their own loop. Okay, but the idea is, let's say the activity of cortical neurons is like correlated and highly related to activity of particular neurons in M1 and particular neurons in higher order auditory cortex. There'll be like a correlation, among those cortical areas that will cause like the thalamocortical loop in M1 and higher auditory cortex, to maybe be active together with this first loop. So the idea is the actual loop, it's not just a tiny little area, it's sort of distributed across maybe multiple cortical areas where, those cortical neurons are correlated, it's, related to each other.
We have time for one more question. Go ahead.
Yeah, okay, this may be a bit more provocative question. So I think it makes a lot of sense in the perspective of trying to explain what we see in biology, but now I hear people from a neuromorphic perspective are thinking about like how to build stuff.
Yeah.
And it seems these are extremely complicated solutions for relatively simple problems, right? To distinguish which of two cortical inputs is stronger, and then to build like two populations representing two different things, having one-to-one matched feedback loops with thalamus and then have winner take all there. It seems like a very convoluted solution. So there has to be more going on why this is necessary, or biology is inefficient, or I don't know, the model is inefficient. So I'm not sure, like, we have some idea like what we could take away from this, like for, I don't know, like, I don't know.
I think if you have like, two possible movements, and you're definitely doing one single task, all this machinery is probably overkill. But I think the important idea here is that this scales up for the whole brain. you just have this common circuit. So no matter how many tasks you're trying to do with the same, robot, you have this common system that kind of always does global motor coordination and has some of these features. So like my guess is that this sort of dynamic might be something interesting to put into your robot if the robot has like many possibilities of like what task could be doing now, versus other things. So if you only have a few tasks, it's sort of obvious what sensory cues trigger the robot to do task A versus task B. Like this stuff is probably overkill. But maybe in the regime of many tasks, this idea might be useful.
I mean, in biology, every single long distance connection is a very expensive concept, right? And so this is kind of like sketching out maybe a minimum necessary circuit in order to kind of like accomplish this.
Yeah. I mean, another idea here is, this is probably efficient compared to building a whole bunch of connections among cortical areas, because it's, kind of a smaller number of connections to thalamus and back, and they're all kind of centrally located. And so, it's potentially a more efficient way of doing this global communication than distributed or trying to do all cortical connectivity.
Yeah, but I mean, an example of why something like this would be something a neuromorphic engineer should take into account is, in the visual system, given their limitations on photons and sensitivity, you want to integrate input so long as your eye is looking at the same place. But as soon as you move your eye somewhere else, you need to turn off, set all those integrators back to zero and start integrating again in a new place. And so one idea is that this circuitry that broadcasts something having to do with movement is useful for resetting the integrators that when the physical stimulus that would drive them is no longer around.
Okay, well, let's thank Michael again. Okay, so okay, can I have your attention? Come on, we need to sit down because minutes of time here. So just to give you some perspective, if you look at what we know about the brain, you could use the last thought, people reported all over literally the entire brain, one neuron at a time. That era is over. And we have a new era, you'll see, in which it's not possible to record from many neurons at the same time, from multiple brain areas. And it's giving us a completely different picture. The global picture looks completely different from what we thought it was just a short while ago. Just back, say, 10 or 20 years, right? So Gerald, his background, he has an unusual background because it's primarily on the molecular genetics and biochemistry. He has an MD as well as a PhD, so he has some medical training. But he also, interestingly, he has he has computational training. So you'll be seeing that he's going to be using some computational tools for analyzing very large data sets from recordings from primarily the zebrafish, where you can record. There's only about 100,000 neurons in the larva, and now we're in this with how to interpret the results, the recordings. So this is something that you can hear about from Gerald and see, actually, not just hear about.
Okay. Well, thank you for inviting me. I didn't hear.
Oh, and he's telling us where you're from.
Yeah, I recently joined the Okinawa Institute of Science and Technology Graduate University. This is a scientific experiment of Sidney Brenner that is basically just had its 10th anniversary. And if you are interested in the place, we're hiring 20 faculty by 2026. So if people are interested in
in a tropical, subtropical graduate university where you're fully funded and grants are optional, this might be yours.
So, open out is Hawaii to Japan.
Yeah, that's right. Good snorkeling, right? Good scuba diving, right?
Yeah.
Yeah.
So, okay, so today I'm gonna come, I think, for left field for everybody except for Aaron Sampson. Because he's already familiar with these kind of things. So everything that I works on low dimensional manifolds. And then okay, so here just to explain the generic concept here. So this is the Lorentz attractor. This is basically a low dimensional manifold that is determined by the flow of basically, if I can get this to work. Okay, so of these three ODEs that you have, and the important point is that they're coupled. And you actually have X depends on Y and X, Y depends on X, Y, and Z depends on X, Y, and Z. So there are these coupled ODEs that determine the relationship between these three variables and the possible values that you set the boundary or the possible values of it is if you run this forward in time, you basically get this dynamical system and you represent this as this low dimensional manifold, which specifies all the possible states between those three variables. So if you actually take this variable X is the project, basically on this manifold, the projection of X is basically what you see on that axis. The projection of Y is on that axis, and you could actually make all three into time series. You have time series, and the time series projected into the space is what it generates this manifold. And this is the space in which I work in. So you can easily go between time series and the manifold, and from the manifold, you can also reconstruct the time series. So
this is a beautiful system, by the way. This is something that also nonlinear control people use, right? It's a nonlinear system, but it's actually a system that you can control in some way because it has the gain terms in it.
yes, and it also, for example, there's like famous method. This OGY method is like Ott, Gregorovi, and York system that is basically pushes it around the bifurcations. You push it a little bit, and basically generally find ways to control chaotic systems. So, anyway, so what I'm going to talk about is a technique that was invented by Jim Crutchfield, but the paper was actually called Geometry from a Time Series. And this is a 1980 paper from this based on the undergraduate thesis of Jim Crutchfield. Some of you might have heard of him. So anyway, so he's a professor now at University of California, Davis. But when he was an undergraduate, he came up with a method to generate basically, if you only have one time series out of that entire system, that system which I showed you at 3, basically you have this manifold as 3. If you have just one time series, you can reconstruct a diffeomorphic. Basically, you can generate basically a manifold that is basically stretched in some way compared to the original one. So you can actually get a lot of the dynamics back. And this is basically what we call geometry from a pencil. I'm going to explain this. This then eventually got, called the Takens theorem because Forrest Takens in 1981 published the proof that this is generically true. So the thing is called Takens theorem. However, it was actually originally first shown in that paper that I showed you before in PRL. So here I'm going to do an animation. It's an easier way to understand Takens theorem, graphically. So here is basically this is the formulation of Takens theorem. So if you actually take a manifold and then you can actually generate an embedding of that manifold in a higher dimension. Let's say this is basically the time series of X. This is the time series of X shifted by a little bit. And this is this amount, the shift is tau. And then you're going to shift it over by 2 tau. And you're going to make these now your new coordinate going to be from x of t minus tau, and x of t minus 2 tau. If you actually do that, what you get is this other manifold. And this manifold is like basically a distorted form of the original one, but it preserves the topology. So for example, you have the two lobes of the original Lorentz attractor. It preserves that. And what is more, every nearest neighboring point on that manifold is preserved. So you only have a stretch form of the original one. So this is the correspondence of the original Lorentz of the with X, Y, and Z. And this is just X and delays of X. So this correspondence that you actually have, this is basically called diffeomorphism, where you have a stretch form of the original one. It's like a topological invariant of the original Lorentz attractor.
And this is proved.
This is proven, and it's proven actually as this was originally there with a delay embedding, and it was actually generically proven for all cases, and the embedding will exist in something called the Whitney embedding theorem. Whitney proved that if you can, you'll get an embedding and a dimensionality that is minimally the same dimensionality as the original one, but by 2n+1, n being the original original dimensionality, and 2n+1, you're guaranteed to have an embedding. So it places a boundary, an upper bound of how high dimensional it has to be for you to get an embedding that is diffeomorphic to the original one.
So if you looked at the embedding, time delay embedding of Z.
Yeah, so the time embedding of Z has a little bit of a problem because from that, if you come from Z, you have this two-fold symmetry. So you're gonna see the shadow from Z, you're gonna have them overlapping. It's still true, but it's basically
You can't see the two lobes.
You cannot see the two lobes either, but they're there, but they're just not easily visible by your eye. But mathematically, it's nothing is violated. So this is basically the formulation of, and then here just to recapitulate, what you take is the original one, and then take one observation, and out of a single observation, and you do delays of the original observation, new orthogonal axes, you can generate a diffeomorphism of the original manifold. So how can you use this? So one uses, for example, here is you have something that, chaos or noise. You would try to distinguish between something that's chaotic or noisy. And here you're going to do a time delay embedding, and I'm just going to show you a quick example of how to actually do this. So you take this, you have two things. I tell you from the beginning, you have chaos on the left and noise on the right. But I'm going to take the first time, I'm calling this X for today and Y for tomorrow. And then I'm going to get a two-dimensional vector that I'm going to put in a two-dimensional phase space where where your components are like X for today and Y for tomorrow for the first time point and the second time point. And then I'm going to plot a dot. And then I'm going to shift over one. And now time point 2 is going to be my new X. Time point 3 is going to be my new Y. And I'm going to do this again. And now you start, if you do this through the entire time series, you're going to start seeing this. If you have noise, you have basically no pattern whatsoever. But if you have something that's chaotic, you're going to see some patterns here. However, for example, if my value today is like 0.75, I have multiple solutions to that. Yes.
When you say noise, it's like Russian, like what are you?
White noise, right?
White noise,
Yeah.
basically like, it's basically like you take the first point and you make a random number generator for the next point, and then basically just.
Well, what if it's band limited white noise?
I mean, anything that is basically, you'll get the same thing no matter what noise you get. There's only one particular case where there's red noise, you're gonna start seeing something. But anyways, let's yes.
So the classification of this is like in the frequency domain, or when you're classifying noise or chaos, because chaos should have a heavy tail distribution.
Well, I don't care about that at all. I care only about predictability. So I don't look at the spectrum of that at all. So because the spectrum doesn't contain information that allows you to reconstruct the manifold. So, anyway, so here, if I had this value here, I couldn't decide here what is going to be my next time point. So here, basically, what you have is called singularity, where you don't actually have a unique solution to this. However, if I take this one step further, and remember, I use like today and tomorrow. Let's say I use yesterday too. Today, tomorrow, and yesterday, and I do it in 3 dimensions. And in 3 dimensions, you're going to see on the next one. all the solutions become unique. So I unfolded a manifold, and now it's basically, I remove all the singularities by doing this. So this is just a practical method that you can use to analyze any kind of time series. So the way you actually make use of it to predict like future states, you take, for example, a particular point that you have, let's say you have one point, and you find this point in the space, and then you find like the nearest neighbors, and each nearest neighbor is going to have a time index. So you project that time index one time step further, and here we actually use a simplex. So the three-dimensional space, you make basically a tetrahedron with your nearest neighbors, and then you predict I want to predict what's the future of this point. I take the nearest neighbors, make a tetrahedron, and then I just said, what are the time indices of each of these vertices of the tetrahedron? And I'm going to project it one forward for each corresponding time step, and I'm going to predict this. The future state is going to be the center of that tetrahedron. So it's basically, you can do it like 2 time steps or 3 time steps or whatever number of time steps you want. At a certain point, if the system is chaotic, then the prediction horizon decays, the predictivity decays because you have a bifurcation, you don't know where it went, and things like that. But for example, if you have a limit cycle, it's predictable, to infinity, basically. Yes.
And the nearest neighbors here are in the three-dimensional space, in this embedding space, not on the manifolds.
Well, it is on the manifold that is from this three-dimensional space from the delays. So basically, it's not the real X, Y, Z, but is basically in the phase space representation of your manifold.
But you don't need to compute what this manifold is to do that, right? You can just take a point in this three-dimensional space and then pick the nearest points in that two-dimensional space, not the nearest neighbors on the two-dimensional manifold in this three-dimensional space.
Well, you pick the nearest neighbors of the point that you have in the dimensionality of the manifold that you reconstructed.
Okay.
Yeah. Anyway, so the typical way that you do is like you build a manifold with your library, and then you try to forecast, like you take half of the time series to make this manifold, and then you try to predict the second half. And that's typically a way that you go about this. Now, the interesting thing part here is like, then you say, okay, the thing that I showed you before, you had with 2 dimensions, some of the points were ambiguous, right? So in 2 dimensions, it was not very good. When you go to 3 dimensions, it becomes much better because everything is unique. But then you start doing more and more delays, more and more dimensions. You start adding dimensions, and with real data, what you start getting is the decay of predictability. So predictability that you see here is like observed versus predicted over mean over the entire time series. You try to predict the entire thing. And then the mean of the observed versus predicted, Pearson correlation of the two, is what you see here. So perfect prediction is 1, no prediction is 0. And then you start seeing that as you add dimensions, the predictability goes down. And the reason this happens is because, let's say you already have this thing resolved. It's basically like, let's say I will try to predict the weather. If I take information from today to predict the weather tomorrow, I have some information. To yesterday's weather maybe is relevant for the information tomorrow. 3 days ago, maybe it's also important. But the weather like 2 weeks ago, to add into manifold, I'm diluting my good information with basically noise or irrelevant information. So your predictability becomes worse. So with this way, you can also find the embedding dimension will tell you what's the natural dimensionality of the original system. So you basically, if you have one time series, you can determine this time series was part of a system that had 3 or 4 non-redundant things. Yes.
And what if you have a system like this cart hole that we like to play with, and it has a 4-dimensional state space or a 6-dimensional state space? Naturally, a measurable state space.
Yeah. So if you do it, probably you come up with something that is going to be between 6 and 13 dimensions in your estimation.
But how do you deal with those different dimensions? Like here in the original Lorentz thing, you just took one, you took like just X.
Yeah.
You did the embedding, but
yeah.
what
So I take one of yours. I mean, here in your case, you know everything, right? you know every part.
No, there are some states that we can't measure, for instance, not
So-
on the particle, but
So
on the.
basically I would take like the time series of every one of the things, and then you try to do this trick. And then you, from a time series of the output of these things, and then you could estimate what is the dimensionality that you can estimate with like 6 different time series.
I'll ask you later, yeah, how you deal with higher.
And then you can actually figure out like, okay, given these that I tried, what was the real dimensionality of the real system? Although I have 6 that I have control, what is the real thing? I mean, probably you could find 6, like I would imagine, because, you know, anyway. But I mean, you could try, you could easily do this. So the Whitney embedding theorem tells you.
Okay, before the, there's overfitting is something that's very common when you're trying to approximate some function, right?
Yeah.
Is this increase in dimensionality in the
Well,
sense of overfitting?
This is probably not overfitting. I mean, you only have 2 free parameters for fitting, which is tau, the time scale, and how many dimensions. So you could actually, I mean, so the probability of overfitting is low because you have very few things to fit with. So, and also the way we avoid overfitting is we do twofold cross-validation, which is the second half of the time series we don't touch at all, and we're trying to predict. If it predicts well,
Yeah, but that's what happens in machine learning. Everybody does that in machine learning. You have tests and training set. And overfitting is a real thing for machine learning, but you're saying it's not.
But in machine learning, you have like 1 million parameters to fit, right? Here you have 2.
No, you can have small networks too. I mean, that's what I'm trying to ask is whether this is completely different from what other people in machine learning do.
Well, it is completely different because you don't, I mean, you're not, it might be the same. You actually get the solution that you have with machine learning might be a low dimensional.
Okay, so this is fundamentally.
But the way you get there is completely different.
Right, okay.
Yeah.
So if you have something that's really pretty high dimensional and you do dimensionality reduction so that your X and Y and Z are, to reduce it to 3D. Does this theorem still apply that you can do the predictability? Or
I mean, the theory says it doesn't have any limitations on the dimensionality you can use. But in reality, you're limited by the amount of data that you have, because as you go up in dimensionality, it increases to the power of the dimensions that you have. So then in reality, like, so the typical example, like if I try to fit a line in 1 dimension with 3 points, which is like almost a minimum. If you try to fit a 10-dimensional hyperplane, for the same precision, you need 59,000 points to fit it. And if the shape is more complicated, then you need more points for that. Yes.
But the variable that you use, this time series, has to have some relation to the whole system, right? I could just add a variable that is just going in a circle all the time and then take that variable.
It's like it's only relevant, that variable, to whatever it's related to. So if there are other things in there that do not have any influence or have like a marginal influence to the other one, then you don't see that. You only see the things that are like sort of weakly interacting, but detectively interacting, between that variable and whatever else might be there. So.
Sorry to divert you here.
Yeah.
Isn't it very sensitive to noise? Because I mean, the parameters are the data.
Yes.
And any noise in that data is going to cause us prediction error. In other words, it won't learn to filter out noise necessarily, right?
Yeah, you don't.
The parameter is the most recent data.
Well, the most recent X number of data as related to the dimensions. But you also use not only that, but all the other state spaces that are around there. So you're not only getting the immediate times before, but all the similar ones that were similar trajectories. So you actually don't overfit as much as you think. And there's a way, for example, if you were to like, instead of taking just the nearest neighbors, exactly nearest neighbors, if you know there's more noise, you can actually use the local hyperplane. If you lose the local hyperplane, you're utilizing the entire neighborhood, and then you have.
I think that'd be worse though, because it wouldn't be robust outliers.
Well, you actually do it basically. We actually have a way to do this. Basically, you take the local hyperplane with an exponential weight that allows you to predict globally the best, and then you take the size of the local hyperplane is determined by you to predict globally. So that's something called basically we call it sequential linear maps. Yeah.
Just from the intuition, it's very similar to Taylor's series, no? Like you have a point, you have derivatives of the point, like rates of change, but instead of having rates of change.
Well, it's not, but the difference is that you don't take just the rate of change of itself. You take all the other ones that are in the neighborhood, not necessarily of itself, but the entire neighborhood in phase space.
Okay.
So okay, so anyway, this is what I explained earlier, which is the Whitney embedding theorem, where the embedding dimension that you find is going to be somewhere between 2n plus 1 and n. So by 2n plus 1, there's a guarantee that you're going to find the dimensionality. So this entire methodology tends to overestimate the dimensionality by 2x, roughly speaking. So anyway, this is just to explain. You can take any of the time series. So basically, and here there was an extension of the Tarkin theorem, which is called the generalized Tarkin theorem. And the thing that Terry was talking about is that if you take the z axis because of the symmetry between the two sides, if you were to look the shadow from that side, for the projection onto the z axis, then you would actually have these things overlapping. So they actually look like there's a violation of it, but there's actually mathematically not a violation. It's just that they're very close and it's hard for you to distinguish. So how do you actually resolve these things? One of the way to resolve is what is proven with the generalized Tarkins is that you can actually mix any number of variables and any number of delays. You can mix them, and it's actually allowed to do this, and you still get a diffeomorphism. So basically, in this case, you actually took y minus tau and z and combine them, and then you can get the two loads back, for example. So this is important because then this some of these ideas went into this paper in 2012 that led me to go do my postdoc with this person who invented this. His name is George Sugihara. He's a mathematical ecologist, and he was like the only graduate student of Robert May. The way he came about this is okay. This is he came about with a method called convergent cross mapping. That's what actually used in this paper. So here I'm gonna show a short video of how convergent cross mapping works. So you remember that you actually get a diffeomorphism. So the shadow manifold of X, what I call shadow manifold, which is the delay embedding, the shadow manifold of the original shadow manifold of the original system M. M you might not know. You only observe X, but you actually have, and remember, every time, every neighbor, every point that exists at M also exists in the original M. And here you also have it for my shadow manifold Y. So you have two variables. And let's say you have these two variables and you don't know whether they actually belong to the original one. So one way you can find this is that there's a correspondent of all the nearest neighbors with the time indices using as identifier of the nearest neighbors. So you can have like correspondence of the neighbors. You could also predict across. So I can say if these neighbors that I use here predict the next neighbors, the corresponding ones should be all the same in the other ones. So you can this allows you to cross predict across two variables. So if you can cross predict across two variables, that is evidence that they originally belong to the same system. So if they belong to the same system, you can cross predict.
Can you expand on that? Like, what do you mean belongs to the same system exactly?
So they were originally along. So here, I'm going to pause here. So basically, like X, there was an X, and there's a Y, and there was a Z that made up the original Lorentz attractor. If you had, then the question is whether does X belong to M, to the original Lorentz attractor or not? Did Y belong to the same Lorentz attractor? Were they both part of the same system, of which you only observe two things?
But it had to be a dynamical system that is governed by some ODEs.
Yeah,
or
the ODEs that you don't know.
ODEs that could also include discontinuities in them.
Yeah. 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 So basically, this is the general concept of how this thing works.
And how is that related to causality?
Okay, so this is so basically like the way it's related to causality is because in the original system, all three variables were causal to each other. So the only way that you can actually find things that have nearest neighbors that preserve across each other is because they were causal to each other. If they were not causal to each other and they were running independently, you would not have correspondence of the nearest neighbors. So there's a geometrical representation of causality that corresponds to basically that you have a diffeomorphic shape underneath that every point that you have allows you to predict across. In a real system, because you have noise and things like that, you're not going to have a perfect prediction. And in certain cases, you don't have perfect predictions because of geometry. For example, just because of the Z, this kind of symmetry where the projection of the symmetry is not good, that you lose some information, then you cannot get perfect prediction, but you're going to get some prediction. So the general idea is that if X causes Y, then you can actually get some from the effect, you can actually cross map onto the cause and then determine whether this thing was causal to each other. All right. So this is just to recapitulate the general idea. If you have the original manifold, you actually have X, Y, and Z at time t. You're going to have X of t, X of t minus tau, X of t minus tau is going to have this neighborhood, and Y of t, Y minus t minus 2t, 2 tau, you're going to have this neighborhood, and you can project from the indices, the time indices of t plus 1 would go wherever it goes, and then there's this equivalent of t plus 1 of this would go somewhere, and then you can predict.
So Gerald, if they weren't causal, you wouldn't get the original manifold. So
Yes.
is the point of this that you can recover whether something that's isomorphic to the original manifold just by looking at the time series in X?
Yes.
Yeah.
Yes, and then you can see if the two belong together.
Yeah.
So, yeah. And then you can account for the ones that you don't know by delays. So a delay is sort of a placeholder for a variable that exists, but that you don't observe. Cool. So there's actually an additional criteria, which is
So, that last statement, does that mean that you can start getting to delays that no longer reconstruct the original?
yeah. So if you have too many delays, then your prediction is going to be worse because you're inferring variables that don't really exist,
Right.
and then you're going to mess up your manifold by adding like extraneous stuff that doesn't really work. Yeah, exactly.
Right.
Yeah, this is a practical. Yeah. So anyway, so if you have a denser map, if you have you have more points, the neighborhoods are becoming smaller and smaller. And if you're becoming smaller and smaller, then the predictions should be better and more accurate. So there's a convergence. So if you actually look at the predictability, it increases, and L is the number of time points that you have. With more time points, you should actually get better predictions up to the point that you actually saturate it, given the level of noise that the system has. Okay, so here's an example from the original paper where you actually do this with paramecium and didymium, and these are the populations of these in an experiment. Yes, I just.
In this case, is Y influenced by X? I mean, what is the red line?
Okay, this is X onto Y and Y to X. So basically, like you're trying to define that whether X is causal to Y and Y causal to X. But if you actually have like basically, if you have, this is what I'm going to show in the next one, where you actually have feedback. If you have feedback, you're going to have bidirectional causality, and you can detect that. And you can detect also which one is the driver and which one is the feedback. So for example, in this case, you can see the blue line is the driver because the effect is more immediate and it's stronger, and it's weaker effect as the feedback, and there's more noise in between, then it's going to take a longer time for it to. So actually, you require more data because the data is less clean. So here's basically just showing that you can actually do it with real populations, and you can actually see that basically the predator has a stronger effect. Paramecium cross mapping of the didinium is stronger because didinium is the predator. So the direction of cross mapping is reverse of what you think of causality. So you cross map from the effect to the cause, but this is how it actually works. So anyway, so you can actually see that the predator has a stronger effect, is immediate.
So what, I'm sorry, what are you measuring?
You're measuring numbers of these organisms in the tank.
Got it.
Yeah.
Okay, that was the piece I was missing.
Yeah. So basically you do this and then you can actually predict basically how this system is going to evolve, etc. You can also determine the
Well, you can just see that. You don't, I see when red starts going up, blue starts going down.
Yes.
And so you can just see the causality there.
Yeah, I mean, this is like a simple example, for you
Okay.
to show that it works, right?
So for you, causality means predictability, really? Like
Yes.
you can predict one system from the other.
Yeah. Yeah. So that's how you find it. But you can also find it through, let's say, correspondence of nearest neighbors in the reconstructed manifold. But the way to practically use it is to do predictability across two variables. I mean, you could just strictly take like correspondence of the nearest neighbors for every point. But it's just a little bit more useful to use predictability. And that's how you determine the product. Because if you think of if something is a cause to something else, there's necessarily going to be some influence of the cause on the effect. And then there's going to be some predictability across that. If there's not a causality, but they can be like weak and they can be combined and can be in a complicated way. But most of those are detectable. So here is basically a graduate student in Song Han's lab approached me that they were having they're using an optrode in this new nucleus that they had in their controlled respiration, and then it's basically they had a single optrode in this place called the lateral parabrachial nucleus, and then that is basically just a fiber, an optical fiber that's placed there in an animal that's transgenic for GCAM, where you can measure like fluorescence, and then you can actually get voltage of the what they call they actually have a thermistor, which is basically a platinum wire inserted into the nose of the mouse, where when the mouse breathes out, it's warm air. So you can actually see the high temperature. When it breathes in, it's cold air. It's like, then you can actually estimate the breathing rate. So every breathing event you would actually record. And then you also have the calcium signal. So if you actually look at the correlation between these two, it's never better than 0.4. It's always like 0.3 or so. The cross correlation, even if you actually, with any delay, the cross correlation is between 2. But if I make a 20-dimensional manifold with delays of that optrode, I can get this observed rate of the prediction of observe. You can see this. I can get to
20-dimensional?
Yeah.
20-dimensional.
But it is an optrode. So basically it's getting like a bunch of neurons in the neighborhood. So it's getting signals from all kinds of neurons. It's not a single neuron. It's a population of neurons. And actually, when you actually look at this, I can get to 97% accuracy about 9 seconds ahead what the mouse will breathe, the breathing rate will be. And what there's more, you can actually do, you can actually use the calcium to predict the breathing, but you can also use the breathing rate to predict the calcium signal, because it actually feeds back. So we actually know this because, for example, when people are, for example, you can actually slow down your breathing actively. And by slowing down your breathing actively, you actually can calm yourself down. So this actually, this nucleus actually goes into the amygdala, and actually you can control emotional states. And basically you can actually control the breathing rate by, actively slowing down. And then you can actually see how this is actually happening. This actually happens to be also the nucleus that is sensitive to morphine. So basically here you can see if I actually have this optrode, I can predict fairly well on this animal. It's like if I add saline, inject saline into the animal, the predictability goes up. So this is the CCM. This is the predictability as a sample size. You see that? But if you actually give the animal morphine, then the predictability. This is the proof, experimental proof that this CCM inference was correct. So, anyway, so the other question is like, can we actually take this and take it whole brain scale? So this is basically the system that I was working with. So I had a neighbor in the South. His name is Shrek Chalasani, and he had a postdoc who was starting to go to Joe Fetcho's lab and using light sheet microscopy to image basically whole brain zebrafish data at single neural resolution for basically a low oxygen escape behavior. So the question is like, how could you actually do this? Well, this is something that you cannot do so easily because the initial data that I had was like 70,000 neurons. That would be 4.9 times 10 to the 9 binary CCM runs. And the calculation at the time when I started this thing, it will take 4,600 years to calculate. So I repackaged this thing, and as first it was in R, so I converted it to C++, redeveloped CCM into also from a supercomputer.
What's ABCI?
ABCI is this AI bridging cloud infrastructure. This is a supercomputer in Japan. At the time when it came out, it was number 5 in the world. It does, it does 37.2 petaflops at double precision. And basically, I ran this, and it took, world's 5th fastest supercomputer 8.5 hours to complete all nodes except 2. 2 nodes were lagging and took 23.5 hours. So I would say roughly like 8.5 hours you can get most of it. I don't know what went wrong there. The lagging nodes and this thing. But obviously this is not very scalable. So you can do this, but it's not very scalable. So actually,
incidentally, that power efficiency, that 15 gigaflops per watt, that's really bad. That's very bad power efficiency, right?
Yes, I mean, but this in the high performance computing world, and the Green 500 was near the top.
Yeah, I mean, it's general purpose.
Yeah, it's general purpose with GPUs. So every node, has, you actually have, it's a combination of Xeons with Tesla V100s. And then,
but just by comparison now, that's AI accelerators are working with, modest fixed point precision are getting like up to like 10 teraops per watt. 10 teraops, right? This is 10 gigaops, 1,000 times better.
Yeah, I mean, this is like this computer came into be in August of 2018. August of 2018 was the one of the things, but we've better since then.
6 years later.
Yep. So anyway. Right, it's about reaching retirement age. So anyway, so I'm not going into details how it actually works. It's just like I redeveloped the software. So instead of being a nearest neighbor mapping, I made it into a bunch of vector distance calculations and number of sortings that could be all done in GPUs instead. So in the end, we basically a combination of hardware and software. The software accelerated roughly now 10,000-fold, and then the combination of everything, you actually have something that's 122 million-fold faster than the original algorithm. So in the practical sense, now I can do it on one high-end server. You could actually do it in 20 minutes or something like that, and something that's much bigger. So. Anyway, so back to the zebrafish. So what kind of things? So this is the causal map between these things. So what kind of things can you find out? So basically this is the causal matrix of every neuron to every neuron. So you can see which one is strongly causal, which like so actually you see this is under hypoxia with the low oxygen, and this is a normal oxygen conditions. You can also see the embedding dimension estimate I was talking about. If you took it under one condition versus the other condition, it actually goes down in dimensionality. So when you actually give hypoxia, when you actually elicit escape behavior, the dimensionality goes down. And you can find a whole bunch of manifolds in there that look like this. So these are manifolds of that you actually find. So here's again to recapitulate. The dimensionality reduction that you actually see, and then you can actually map where the dimensionality reduction occurs. Here, I'm going to skip that one. So basically, you can see the part that becomes more yellow is the part that actually the dimension goes down the most. For example, the optic tectum doesn't do much. The forebrain doesn't do much, but for example, the motor areas, because the escape behavior stereotype becomes more simple, therefore you see things like the dimensionality reduction. So, okay, so here, just to simplify things a little bit, just to show it, I took like the 154 representative behaviors that you actually see in the time series in entire way. And actually, what you can see that under normal oxygen conditions, you have many these clusters that meet, but everything collapses into sort of like one big cluster when you actually have the behavior becomes much more simple when you actually go into this escape behavior. All the other ones kind of got, because when low oxygen, because that's a priority for the fish, I just want to get a
hierarchical clustering of behavior on the two sides?
no, it's optimized for each
sector.
yeah.
Okay.
So to find the correspondent ones, you actually have to go and take them out individually. But this is just to get it. So okay, so I'm actually interested in something else. Is how do I get a summary of everything, and how do I make sense out of this entire thing? And one thing that I want to find is basically what are the things that integrate information in this entire business. So what I want to find is basically where CCM is much better than correlation. So actually take the CCM matrix of the causal matrix of everything against everything. Take the correlation matrix, the Pearson correlation of the entire time series, and then look at the difference, what I call row diff. And from that, you can actually get columns, and then you can get rows. So the columns means that this neuron here is receiving inputs that are nonlinear for these rows, and there's basically this neuron's integrating information from all these other neurons in basically which are not correlated. So this means that you're actually this is basically where your nonlinear predictor is much better than your linear predictor. So you're actually trying to subtract one out of the other. So this is the specific example now for normoxia and hypoxia. And here I focus on this one. And this one actually I chose because it actually maps well to the overall motor activity of the fish. And you actually can generate, you find 3 neurons here, and you actually map it to the overall motor activity of the fish. And when you actually do this, you actually get this manifold. So these are 2 specific neurons, and this is basically the overall motor behavior of the fish. So if you actually map that, you can actually predict the escape behavior of the fish and classify the type of escape behavior. So basically, if you actually see here, I don't know why the line doesn't show up here, but there's supposed to be lines connecting this. I don't know exactly what it is.
If the contrast is too high of this, of the dreamer.
Okay. Well, anyway, so if you actually were to make a mask here, you can actually predict half a second ahead before the fish actually makes a motor bout, and you can actually tell which type of motor bout it does. And here, just summarize the entire behavior of the fish into 3. And one of those neurons is something that David Tank found previously, that is basically a vision-based integrator. And then, and the other neuron is a new one that's in the cerebellum. I don't know exactly what it does. At this point, I'm not really looking into this. Is a technology development at this point, and then I'll get into the biology later. But right now, I'm more into it. Actually brings to this, I think, important idea of correlation not being causation, as many of you know. These are 2 things that are correlated. Yes.
Just a quick question before. Did you choose specifically these 2 neurons, or did you take them randomly?
Oh no, they came out from the data. Yeah. I didn't choose them.
Column.
Yeah, from that column. Okay. So that column, then I clustered them to see the ones that are similar. And then within each cluster, the things are nearly identical. I picked the ones that are the best predictors, and that's the general criteria. Okay, so most of the time people think things in this way, especially in biology. Sorry, oops. So this is not how I was taught as a biologist to think. You first look for things that are correlated, and then you try to do an experiment and determine what's causal. But from this, I already gave you a sort of a little preview of that there might be things that are causal but uncorrelated. And this is not a very familiar concept to most people, but I think it's a very useful concept, and it's very useful because, for example, if you think about it, when you integrate a lot of information from a lot of places, each of the inputs are not going to necessarily correlate with the output, but it's the combination of all the inputs that are positive and negative, and you somehow integrate it in a nonlinear fashion that comes out. That's what these manifolds capture. The manifolds capture basically the little or partial correlations. You could think of it as your partial derivatives that are state dependent, in which the way they integrate things. So if you actually do that here, actually, I'm showing this. Okay, so anyway, this is, I don't know where this is collapsed. Okay, this is supposed to be a movie of fish swimming and then a sea lion swimming there in La Jolla Cove. And there's a movie there, but if I were to do the standard statistical way to analyze this, if the system is a truly dynamical system, I'm taking 10 frames of movie 1, 10 frames of movie 2, overlapping them and trying to find mean and standard deviation of it. This is what you're doing when you're doing an experiment. If the system's dynamical and you try to collapse it in this way, this is what you're seeing. It's not that you don't see things, it's that you're missing the point. So here I show you, now I'm going to switch gears into a different system. This is basically cell cycle in yeast, where I took basically sequencing 57 time points across the entire cell cycle of yeast. And I took, I got all the, and I found in this group of genes here, 3 genes that are completely uncorrelated. The correlation coefficient between these 3 genes are 1. That's the criteria. At any lag.
Yeah.
At any lag. They have no visible correlation at any lag. Now, if I were to plot the correlation coefficient, for example, of switch 4 and clone 3 is 0.08. Your linear fit is terrible. But now if I take, and we connect time point 1 to time point 2 to time point 3, etc., etc., now you see on the next slide, you actually have a very nice, coherence in the whole thing. But here we actually have trajectories that overlap. The trajectories overlap, you have singularities. Those are the places that are ambiguous, you cannot predict. So I'm going to add 1 dimension. I'm going to add 1 more gene.
You need 1 more lag.
Not 1 more lag, 1 more gene. I could do it with 1 more lag, but I can also do it with 1 more gene. And 1 more gene is more meaningful because I know what it is, and I could do something about it. So this is the new manifold that you get. This is the raw data. I didn't do anything to it. This is just plotting them against each other. And now you see all the singularities are gone. So here I have 3 things that are uncorrelated, and I predicted them to be causal to each other. But this actually is part of a thing that actually has 4 dimensions asked by computation. So there's actually a component that's called V5 that is connected to all the other 3. Now, how do I know this is here? And I show here V5 as the intensity. You can see the intensity of the color. That's the same thing that I showed you in blue earlier, but now I show you the intensity of V5 as a function of all the other 3. And you can see this. Now, so I'm going to do an experiment. If these things are causal to each other, let's say V5 is causal to the other one, I can manipulate V5 and I should expect the other ones to change. This is the way I know that I'm not having here a mathematical fantasy of some sort. And
So this is a perturbation experiment.
There's a perturbation experiment. I overexpress V5, and if I overexpress V5, I should see a change in the trajectories. If there's a change in the trajectory, I would have experimentally proven the existence of causation without correlation. And this is what you see. So the blue line is overexpression of V5 superimposed on the original manifold that you actually have. So this is the experimental proof of causation with the correlation. There's several things that are interesting. So one thing is when V5 is naturally high here, it doesn't change a whole lot. But when the levels of V5 are intermediate, the system is the most sensitive. And also when the trajectories are more separated to each other, those are the areas that are more sensitive. So you can use that as a criteria. Although you don't have the ODEs, you can tell from the geometry which area is going to be more susceptible to perturbation. Because you actually can determine the local stability. Anyway, so this is basically just to show that you can improve it. And in general, you can actually do this at 70 to 80% experimental confirmation of these things. So now I'm going to go one step further. And this is the final thing I'm going to show. You can actually take this a step further. So this entire concept, of using manifolds from real data, you can actually do something that I call downloading a brain to a robot one behavior at a time. So how do you do this? So you actually obtain whole brain neural activity, a single cell resolution perfectly, but you don't always have that. And then you extract all the relationships using CCM, convert the relationships into something that can be transferred to a computational framework that can generate the actual behavior of the animal. So that would be my definition of downloading the behavior. And then you can test the performance against the real thing. So the way I did this is using some of Terry and Ralph Greenspan's data, with data that was taken on light field microscopy. The fly was on a basically head fixed. So you actually look at activity in the brain with GCAP with this light field microscopy, and you actually have the fly on the styrofoam ball where you actually record all the movements of the fly on the styrofoam. If you actually look, so all the areas of the brain, because you don't have single neural resolution there, were just separated by ICA. And you came up with 80 ICA areas, and then you do CCM of this, and you have manifolds like this kind of thing. These are just a delay embedding manifold.
Well, why do you have to do this? Sorry, this is very fast. Why do you have to do this ICA first?
Because we have to determine which is the area, your ROIs. Basically, what are the areas that have essentially the same information? And because you don't have single neurons. If I had single neurons, I would do it single neurons, but okay. I don't have single neurons here. So basically here is I get what types of activity.
So this is some sort of summed population.
It's some population. It's basically a mass of a part of the brain. That is like it's defined that they're highly correlated.
Okay.
Highly correlated areas masked together.
Okay.
So are these ADICA areas mutually exclusive, or do they overlap?
They're mutually exclusive.
Yeah, okay.
Yeah, they're all mutually exclusive. Yeah, they're not overlapping. Yeah. So okay. So if you start with this, now the other thing that I have here, remember, I have the movement of fly. So I can have the forward speed of fly and the left-right speed of the fly. That's another thing that I have with this entire thing. And that's the other thing that I have recorded because I have the movement. So I actually have sort of like the behavior. And I'm going to actually do the dimensionality reduction. No, I don't do that. Sorry. If I determine the embedding dimension for each of the manifolds from every single from all the 80 areas, and this is a distribution of it, I have nothing that's linear. 0. Everything is nonlinear. And the lowest dimensionality I have is 3. And the maximum I have is about 10 in this behavior. So there are individual areas that have dimensionality that goes as low as 3 and as high as 10. And this is the distribution. The median that you actually see here is about 5 to 6. So, but as you can see here, if I took this column, these guys are very similar. This is the causal interaction matrix of every area to every area, plus the two left-right movement and forward movement. And you actually can see, I could actually summarize this entire thing as one, because they essentially have sort of the same information with respect to their relationship to the entire network. So you could actually summarize this, but what is more, you could actually convert those into manifolds. So the same thing that I showed you earlier, I had the CCM matrix, the correlation matrix, and I subtract one from the other one, and actually the absolute of the correlation. So it's basically not, so negative correlation will be turned positive. You actually have these, now you just sparsify the entire thing in terms of the as much possible causal but uncorrelated. So you actually have this here now. As much, this is what we call row diff. You subtract one together. It's a very crude way to do it. There are better ways to do it, but this one works. So now the idea is basically I'm going to construct a manifold that predicts the other manifold. So how do I do this? Let's say, for example, I want to look at forward speed. I'm going to have time series 49 that gives me a predictability of 0.394 of the forward. It's not very good. Then I have another time series 56, gives you 0.37. But these are the best ones I could find that allow me to predict the forward speed of the fly. But let's say, so I'm going to take this. For example, I have a manifold that, let's say, to predict the global movement, I'm going to have forward, left, right, and this time series 54. Now, what I'm going to do is I'm going to build another manifold. This tells me the relationship between forward speed, left, right speed, and one particular brain area. Now, I can build another manifold that gives me the best possible prediction of forward, and I'm going to feed this information of forward into this one. So I'm going to check on the, on the row diff matrix, and I'm going to do this iteratively, and I'm going to get the best manifold that I can make that predicts time series 54. And then you keep doing this and try to use up all the data. Oops, sorry. So basically, you try to use up all the data that you have. And sometimes you don't have all the observations, so sometimes you have to throw in a couple of delays. So when you actually end up doing is, this is the algorithm. Basically, you take the row diff matrix, you try to make using an acyclic directed graph. So basically, at every step, you don't have feedback into the same manifold. So you make a whole bunch of manifolds that inform each other of whatever. And how do you link the manifolds? The manifolds are linked by sharing the same time series. So sharing the time series will be the edge that you actually generate, because the edge that you generate is basically a shared time series in that manifold. And then you actually do this, then you can actually project it forward. You can actually take it and we said, I'm going to predict one step forward, walking along that trajectory on the manifold. And if I move this forward, it carries on to the entire network. So if you do this entire thing, so for example, I generated this one that basically goes into left, right, and forward. And this is basically you have like, you have all the manifolds that you could generate with this data set. So these are, each of the nodes that you see here is a manifold that predicts that particular ICA component, the time series. So this is the way I link everything together. So you have an entire network, which is a network of manifolds, and the network is linked by shared time series between the nodes. So you can use this to predict. It predicts fairly well. This is observed versus predicted for different. But what is more, you can actually generate behaviors. And if you take a single manifold and try to generate behavior by walking in one step forward, you end up in this. So this is basically forward speed of the fly. It goes bounce off running and then stops, running and stops. This is forward speed. If you keep doing this, it collapses. Because what happens like on the manifold, you find a neighborhood and gets trapped in that neighborhood, and then you can never get out. But if you actually do this with the entire network, this is what you get. So this is basically the training set, and this is artificial generated data. This is done for the forward speed. And I'm going to show you, this is a brain area. So this is a particular brain area, time series 49. And then you can actually generate things that look also similar in dynamics. Here, you look at a spectrum of forward. So I can say like, this is basically the real behavior spectrum and the manifold network generated, the manifold network generated spectrum. And you can see they're kind of a little bit similar. They have the same kind of decay. And so it's not just by eye they're similar, the spectrum is similar. So you can do it for everything, of course. And this is like a thing that we try to do also. So this is the original movements, like force grain to the time scale of the GCAP, because the actual movement is faster. So actual force grain it to be the same time scale as the GCAP. And this is basically the phase portrait of all the movements of the Drosophila on forward speed and left-right speed. So when it stops here, then runs forward, it's faster, go to the left on that side, and it goes to the right on this side, and things like that. This is what I generated from the same thing. You just run it forward and see what you get. But if you, let's assume this thing was Markovian, and the movement itself, the running of the Drosophila is able to predict the behavior because it's Markovian. So every step, so I make a manifold just of the time series of left, right, and forward, and any number of delays, and optimal number of delays, and I try to do the same thing out of the single manifold, and this is what you get. It collapses and it generates this kind of thing. So basically, this tells you that movement alone is not sufficient to generate realistic behavior, walking behavior of the Drosophila. You need to have the neural data. You need brain data to generate realistic behavior of this. So this was the next thing is a little bit surprising. So in the withheld data, there are these long pauses. We didn't have any long pauses in the training set. And the training set is only 600 points. So here you actually long pauses. But we actually generate this thing. We could generate these pauses. So it seems that the structure you actually have in there has some inherent properties of plausible things that you can do that you don't explicitly have to have present in the training set. And then we try to find this basically a way to basically validate it, and then to have a metric of success, and the metric. We generate a whole bunch of artificial data, and when we generate this artificial data, we do an embedding of the artificial data and see how well can this embedded data predict the withheld data. So this is a way to, how realistic is this? So this is a metric of realism that is not strictly by that spectrum that I think is a little bit hokey. This actually has a, this is really predicting something, So anyway, so this, I'll go back to the metric again later. But here's basically, I just generated a bunch of other things to compare it. So basically, how realistic, we actually generated other things. So basically, this is a vector autoregressive model for the same data. This is basically k nearest neighbors kind of data. And the most interesting here is this one. This is the flat embedding. The flat embedding is what I call as data. You take all the data you have there, right? 80 time series of all the 80 areas of the brain. I make an 80-dimensional manifold. It should contain everything, and let it run. And when you run into these infinite repetitive things. So it keeps going into infinite cycles. It gets trapped into one thing, into one mode. It's deterministic, and it runs in these complicated circuits, but it runs forever. This thing is very good for prediction, basically, but it's not very good for generation. So you can make an 80-dimensional manifold. It will work very well for predicting the next step, but it cannot generate. So here's just to see the output that you actually have. So this was the true data output. This is the generative manifold. This is the left, right versus forward speed. This is the left, right, forward alone that you generate in this run. And this is the flattened network. It just generates this kind of thing. And anyway, this is just to show you how it performs.
But the k nearest neighbor doesn't look too bad.
Well, but it collapses. So actually, the k nearest neighbors is like.
Oh, I see. Okay.
It does this for a little bit.
So
It does this for a little bit, and then it's just flat.
So just looking at this butterfly picture, it doesn't give you.
Yeah, because
it doesn't tell
you
you
don't
what-
see that
what's
time,
being
that's why I have to showcase things, because otherwise you cannot see that. It actually collapsed and went to one point and just stuck there. So this is across every single node. That thing that I told you that we say using every node to the prediction of the every time series and then compare it to all the other ones. So the blue line is basically, I mean, the way I show the blue line, because this is every single brain area and how this is like perfect correspondence would be 1, no correspondence would be 0. And this is like the yellow is the k nearest neighbors, the red one is the vectorial regressive, the flat one is the gray one. And in almost every case, for every single brain area, the GMN, the generative manifold network, is better. And this is the same thing for the spectrum. So anyway, so can we actually implement this at whole brain scale? So we have, at the time, my fish was basically embedded in agarose. I didn't have actually motor output, but given that limitation, I actually had that CCM across the entire brain. And you can have like left and right motor neurons that you infer pairs of motor neurons, because I can see those. I try to infer, can I use the entire brain activity to predict the outputs of left and right motor neurons as a surrogate of real behavior that I couldn't measure. So all I can say that up to this point, for left and right motor neuron pairs, I can actually generate realistic outputs. The orange stuff is the output you have, and it seems to generate realistic outputs. And now I'm collaborating with Misha Arens, who actually has real motor output to do this thing at the whole fish brain scale.
Could you put back the movie? Okay, so I think what you're saying.
Which movie?
Okay,
Oh, yeah, that's fine.
well, the rotating.
Yeah.
Okay, so we heard in the last lecture about this big motor signal that goes to the brain. I think what you're saying now is that by looking at the neurons that would have given the motor output, you're seeing at that moment the whole brain led, right?
Yeah.
Okay, so let's watch it. There.
So yeah, those big, the big motor, the motor bouts that you actually have,
Right.
actually activate the entire brain.
Yeah. So that we saw that in the fly too, by the way.
Yeah, and yeah.
When you walked, okay, it was really interesting. When as soon as it walked, the whole brain lit up. There's another behavior, which is grooming.
Yeah.
That was much more localized, and it didn't have that character. That it was.
When you have these large motor bouts, the entire brain lights up. So it's just not in the motor areas. This thing actually spreads over and it actually echoes a couple times. So you can actually see the same event kind of echo a couple times. When it actually, you can see it, in the forebrain, you can see it, even in the visual tactile, you can actually detect it. So there's motor signals, albeit weak and sparse, that actually show up in the visual tactile. There's some kind of feedback. You somehow needs to know that it's moving. Anyway, so this is also a little bit, this is like a further development. The point here is to show that entire thing that we actually had here to generate that generative manifold network, we can actually compress it much more to actually perform just as well or even better if you actually compress it into many fewer nodes that actually eliminate redundant information in that network. So if you do that, you can actually generate something that performs just as well in terms of the behavioral output. And that is basically applying a different way to compress the data that I call causal compression. That's where that row diff that I think showed you, that's subtracting the correlation from the CCM. But you can actually go further, apply additional criteria for convergence and an additional criteria of minimal mutual information. So if you actually do that, you get something like this small. But the advantage of having something this small, this becomes very explainable. We can actually tell what brain area is really controlling the other ones, and the other guys, I can tell you which ones are redundant to that one. So you don't lose anything. So you have this knowledge at single neural resolution, at ways that I can tell you which one should I stimulate to change which one and how the effect should spread. So this is the thing that, and it has no latent variables. Every single thing that you have this corresponds to a neuron, to a brain area, or to a behavior. There's nothing abstract in this thing. Anyway, so this is basically the summary. So this is a new method that generates embedded representation of brain and neural activity, can reproduce realistic walking behaviors from recorded neural activity and Drosophila. We think that we probably can do it in fish too, at single neural resolution. We're not completely sure because we don't have that yet. But and then it seems to have emergent properties, and it can be done with brain-wide activity. And this is the people that I work with on this. This is the Frank Chalasani, who I started the fish stuff with. The actual algorithm I coded over the I designed with a undergraduate student, Cameron Smith from UCSD. And on the previous high performance computing thing, I worked together with my friend Keiichi Takahashi, also the algorithm, but he's an expert in HPC. And then also with Joseph Park from the CTBTO from the United Nations, the people who monitor nuclear explosions, that we work on this time.
Okay, so suppose you did this with a mouse brain.
Yeah, so right now it's like we don't have observation of the whole program. But I think it's like we could try to do this with, like, say,
But you know, could you do it with the subset of
yeah,
the neurons?
you should be able to do it,
right?
yes. Right now, it's like we have, like, for example, that was a Matteo Carandini and Ken Harris data set from NeuroPixels. That's like 50,000 or so. That's well within the range of what we can do.
Okay, any questions? Go ahead. Well, I was pointing at you, but if you want to let someone else go.
Just about this reduction of complexity, like the last slide you showed, would that mean that you can create like a virtual network that would simulate exactly the behavior of the fish?
It simulates things that are consistent. When you run it forward, it behaves like by all criteria that we've found so far. It does realistic things. Actually, I've done it with the freeway traffic of Los Angeles. I've done the same thing, and it works also. So I can actually get the traffic jams at the same time. Basically, it does the weekends not having traffic. That all comes up. So that works like that. It's not necessarily limited to neural data. There's any nonlinear system that has basically it has a complex network. Yes.
Good.
Two small questions. You mentioned before you were trying to validate the predictions in manifold network.
Yeah.
By taking the prediction you made,
yeah,
embedding them, and using that embedding to predict
real data. Yeah, real data that was withheld.
Right. Okay. Yeah. So because you couldn't use the prediction in the first place because it was withheld, or what's your?
No. The prediction. Only use half the data for the predictions. Yeah. So anyway, the predictions are a different thing. Here we're generating data. We're not predicting. So
but you're verifying whether the generated data holds up to the actual measured data, right?
Yeah, exactly.
So my question basically, why the extra embeddings there?
Oh, because if you don't embed, if you embed it, then you can do all the typing theorem that basically you can do cross mapping. So basically you can do cross mapping of the real manifold for the fake one, that artificial manifold. So basically you're trying to see, like, do they actually have similar structure? If they have similar structure, we should be able to predict using the same kind of geometry.
Okay, so and next step to that quickly will be like to understand that embedding and actually gain insights on the actual differential equation, the actual ODEs, Because with the mess, it's a bit of a black box, right? You don't know, you can't study the dynamics from the first principles, you just predict something.
No, you can predict something, you have the geometrical representation, the bounds, given the data, but you don't have the ODEs at all. If you can come up with a way to ODEs, that, for example, Steve Brunton and University of Washington has ways to generate ODEs out of manifolds and real data. With that, if you use that and it works, great, but it requires a lot of data. So it's difficult to come up with that.
Well, I think the beauty of this is that with limited data, you can detect a nonlinear dynamical system, which is kind of the core. It's not all the details. You don't even have access to a lot of the details, but it means that there is underlying this very complex pattern of activity. There are these low dimensional manifolds which capture most of the behavior, right? That's what you're saying. Which is really quite remarkable. I wouldn't guess that.
Yeah, it leads to the question of whether you can construct experiments hoping to take advantage of this.
Yes, so this tells you which area is doing what. You can this connects
and
directly
you
to
can ask the question, you know, you already did it actually, where you play around with a couple genes and see how they're related.
Yes.
Well, you know, it didn't work as well when you had only 3 and you had to say, okay, there must be another gene that if this is going to work, you need another gene that's going to contribute to this.
No, I could have done with any of them.
No, I understand.
Yeah.
But you got the sense that you needed more.
Yeah. I mean, this is I got a sense because I found it computationally that there was a 4th gene in there.
Right.
So basically, like, although at 3 dimensions already unfolded, at 4 dimensions, I can guarantee you it's unfolded too. There's no singularities in 4 dimensions either.
Right.
It's just that it's difficult for me to show you in a presentation on 4 dimensions. Otherwise, I'm showing you in 4 dimensions.
Okay, I'll say one more question.
So how far is the prediction method? Like what determines?
That depends on each system. It's different each system. So that's basically if it's chaotic, it's like that. If it's like if I talk about the orbits of planets and I embed it this way, I can predict like billions of years.
So could you use ensemble methods here?
I don't like ensemble methods in the sense that ensemble methods try to tend to get averages of things, and that as I showed you that if you average the things and then combine them and find means of things, you lose the whole thing.
So, okay, so remember the picture of the video. So I've seen it. It's really quite nice. So the fish are moving around like this. You average and just get a big blob, right? Because you're averaging across different behaviors and changing behavior. And in a sense, you know, when you record from neurons and do spike triggered average, or if you do post-stimulus time histogram, you're averaging across, and it'll blur out effectively the real details of behavior of what's really going on in the brain, generating a particular behavior. So I this is a new way of looking at the data.
I mean, there's other ways to do prediction too, like recurrent neural networks, Gaussian processes, there are a lot of different methods. And so we're particularly interested in those for control systems, where we learn the dynamics from data.
Yeah.
And it's hard for me to judge. I mean, it seems kind of interesting to look.
Yeah, so basically the big difference here is like the whole thing that I showed you for the fly brain, 600 time points.
600 what?
Time points.
Time points. Yeah.
That was
many methods claim that they only need a small amount of data, like Gaussian processes are claimed also to use a small amount.
I mean, I'm not saying that like there are many ways to do it. It's like this is just an extra one. I'm not saying that any of those don't work or anything like that. They obviously work. People use them forever and they work. In the end, it's like which tool is best for which circumstance.
And what is the inference cost here compared with other methods? In other words, if you want to do this stuff in real time, you have a bunch of data now, you know, from
right now that I think like the way I'm imagining how to do this is like I generate the models, I put an FPGA, and basically the actual manifold that you get out of it is going to be an FPGA, and the predictions, if you look anomaly detection or any prediction ahead, that's going to be implemented in
FPGA logic circuit.
Yeah, basically like you're gonna you just have the entire structure of the manifold in an FPGA hard coded in there.
Big FPGA,
Yeah,
or little, depending on.
like Xilinx, it works.
It's like kernel method, right?
You know more about it. Yeah, you should be asking this question.
So you're using for multiple
Yeah,
and you can multiple manifolds by
exactly. So you can actually do it, you can actually do that, you could, if you are interested in a particular system, then you can actually use just one manifold. You don't have to have the entire network.
Okay, well, we should break down this. Thank you.
Moein from Cortical labs and Monash University. And they've recently put out this really fascinating bit of work where they took some brain cells, threw them on a disk, made them take phones, got an incredible amount of PR and traction and money. So safe to say they're doing pretty damn impressively. But hey, don't let me give you a talk for you. We're take it away.
Thanks. So hi again, everyone. I guess.
And just talk up a little bit.
Okay.
Wait
Yeah.
for the appearance.
Yeah. I guess we've met almost everyone here, but if we haven't met yet, I'm Forough. We're coming from Melbourne, 28 hours flight. But then glad to be here, and I'm happy to talk to you about a little bit about like the background, basically the paper we put out, how we built our system, the preliminary results we've got, and also Moein is going to also talk about a couple of ongoing projects we have working with this data set. Well, none of us are among the founders of Cortical Lab, but we were lucky enough to join them very early on in their second year, I guess, of working. But we could, I guess, safely say that maybe one of the main motivations behind the entire idea was this speaking intuition that maybe humans now start to think that technology like AI has run ahead of us in terms of intelligence. But then we wanted to somehow prove that our brains are still probably more capable of AI in so many different aspects. Well, without getting too philosophical, we're talking about intelligence here, and we know, or we can agree on, that we need some form of intelligence to be able to distill meaning from different phenomena, and actually being able to make intelligent decisions using more data faster and in a more accurate way is one of the main goals of AI and machine learning. So everyone basically is in search for intelligent computing system these days, and there are obviously many approaches. One way to categorize all of these efforts would be to talk about machine learning and AI, quantum computing, and then of course neuromorphic computing. Just looking at the half empty of the glasses right now, we know that, for example, in machine learning, we face issues like high power usage, their inflexibility, and what we call catastrophic forgetting when they're facing different or new tasks. The fact that they require very large sample sizes and very large training times, and also they have higher error susceptibility. Then talking about quantum computing, not that I know a lot about it, but at least we know that it also needs extremely high power usage, and it is isolated from the external world, hard to verify, and also not really adaptable. Then coming to us, the neuromorphs, we should accept that we also have challenges and limitations like the limited error correction or the variability between different circuits that we have, and also we still do require some sort of programming. But at least we know that the brain is the most advanced intelligent system available. So we thought, why try to model it if we could actually harness its capabilities? We know that biological neurons have this innate learning ability. The neurons within the brain are actually capable of continuous and flexible learning of novel and also distinct tasks. These biological neural networks have extremely lower power consumption in comparison, and also they can maintain their functions when facing different variations in the environment or even facing some damage. So just the certainty that the brain is capable of being intelligent is what is actually lacking in the other categories of efforts to make or generate an intelligent system. So our question changes now from if to how we can harness these computational powers of the brain. So basically, we wanted to build a synthetic biological intelligence, or what we call SBI. In our effort, and when we were designing our system, which is called Dishbrain, and I'm going to talk about its detail in a bit, we basically, I should mention that we basically benefited from a combination of obviously all the hardware and software developments in recent years, which made us capable of designing a real-time closed-loop system or an embodied game environment. Also, the more detailed and more recent, quite recent theories of understanding how the neural systems optimize their behavior to become intelligent, and also the synthetic biology advances and the fact that we could use human iPSC cells to create scalable and specific neural populations for our purposes. Okay. So now, just to talk about what we have, what's this system that we're talking about? Biologically, we have these cultures of neurons or these tissues, which are either human iPSC cells differentiated into neural phenotypes, or we also have primary cortical neural cultures from mice. And then obviously all of these neurons are then plated on these MAX1 chips. These are high-density multi-electrode arrays from Maxwell, which is a company in Switzerland. We are making our own chips now, but obviously that's a work in progress. But all of the results that we're going to present to you today, we're use the data that we gathered in Maxwell. So just to validate that what we have here are actually live biological neurons and the cultures are actually alive. These are images after 80 days, obviously a sample culture. The contrast isn't very nice there, but using different stainings, we confirm that we have a high proportion of neurons. We have significant axonal branching, and we also have a substantial spreading and dendritic interconnectivity.
I have a question. Gerald Pao had a big trouble when he was culturing these neurons that the neuron cell bodies wouldn't stay where they started. They would just clump together. And he couldn't find the cell bodies within these little cups, these little glass cups with holes coming out, so the neurons were forced to grow dendrites to connect to each other. What did you do about that? How do you stick them so they don't move around?
I wasn't part of the biological team during culturing, no, but Brett is going to join us after this, so he will definitely be able to answer that question. Yeah. So then again, we did other types of stainings. We confirmed that there is a significant number of higher cortical layers, like cortical layer 2 to 4 neurons. We also did confirm that we have glial cells here, but we didn't detect any dividing neurons or cells. So basically, what we have now is this quite dense interconnected culture of neurons, and they could actually survive to almost 6 months, and in some cases even above that.
But that's remarkable, right? This tissue culture technology has advanced tremendously.
Yeah, 6 months was very surprising for us as well. We've worked with Maxwell chips and also MCS chips. We've realized that Maxwell ones actually are capable of staying alive for a longer time.
So less toxic for the neurons.
Yeah, exactly. So the Maxwell ones reached 6 months, but not MCS. But then also they were susceptible to infections.
Yeah.
So you should.
Yeah, that's right. You don't have an immune system, right?
Yeah. Yeah.
Maybe we'll come to it later, but how about the environment? Like where you place the chip? Does it have to be a very well-controlled environment?
Yeah, just very media inside the well, like yeah.
Did you have an experiment of that which you use like human iPSCs, and everybody tried to go to 9 months where they actually mature?
9 months.
Like 6 months is they're still immature, like at 9 months they undergo a transition to mature.
Yeah, no, we don't have any 9 months olds yet, like that they have stayed alive, but all of our efforts right now is like to be able to keep them alive for longer durations of time. So as soon as we are able to do that, definitely that would be very interesting.
And what are they fed?
Inside the media, like the actual composition, I really don't know the details about that.
What is that?
It's called a mixture of two supplements that are combining a whole bunch of vitamins and growth factors. And people used to have either N2 or B20 supplement, and some people have that you combine the two actually works better. So now it's generically used for like that.
That's the super sterile too, right?
Yeah.
Have you guys started working with organoids instead of just?
That's futurist that we're now trying to work with organoids. So far, it's just two-dimensional. This is again just some sort of validation that we're looking at the electrophysiological activity of different types of cultures. We have the top row is a primary row that the cortical cells in time. You can see the obvious increase in their spiking activity, and then we have the human iPSC cells differentiated into cortical cells and NGN2 differentiation of human iPSC cells, which gives rise to multiple neuronal phenotypes. And in all of the cases, after a certain time point, we see this significant increase in their just spontaneous activity.
That's GCAP.
What is that picture of? That's imaging of activity?
Yes, no, these are spiking activities. Yes.
Spiking activity.
Yeah.
So this from GCAP?
Yeah. So then we put out this paper, which I guess a lot of you have already seen in Neuron, just talking about the entire system, how it works, and how we kind of communicate with the cultures, and the environment, the virtual environment that we use, and we kind of embody the This is fun, just to become familiar with the game. And now the more exciting parts, I'm just going to talk to you about the setup, how we actually managed to do this. So we had these dense neuronal cultures. We kind of integrated them onto a multi-electrode array, which I mentioned to you. These arrays, they have like 26,000 electrodes channels, but we don't use all of them. In this paper, we only use 1,024 of these channels. Basically, what we do first is that we predefine two separate types of regions here. One we call sensory region, which is up here, which consists of almost 600 channels. And then the others, which we call motor regions, these are almost 400 channels. And then the arrows you see here is basically for the motor regions. Well, these are the regions we only record from, and we kind of interpret how the paddle is moving by comparing the activity between different parts of these motor regions. Obviously, this layout is chosen by us and defined by us. We tried so many different layouts in terms of which region of the motor, which sub-region of the motor region would be responsible to move the paddle up or down. And this is the layout which works best. So basically, we provide stimulation to only 8 of the electrodes in the sensory region, and then we report from everything. And I'm going to tell you about the simulation, the feedback, and all of that. So this is another like kind of schematic of the electrodes that we have. Those crosses are the 8 stimulus electrodes that I mentioned to you. What we feed the culture basically is the information about the ball's position. So the ball's positions in X and Y axis. Its position in X axis is rate coded using the frequency of the input. The frequency changes between 4 and 40 hertz. So as the ball moves closer to the paddle, the frequency of the input increases. And then its y-axis location is actually encoded in a kind of a not a continuous manner. So we only have these 8 electrodes. So depending on which section of the culture the ball is, only one of these stimulus electrodes is activated given that specific frequency.
One ties to 80.
1 to 8, yeah.
Here you can use one of the figures that are.
Maybe if you have questions, we're gonna come back to this again or in another part of the presentation. And then we record from the motor regions. We compare the activity of the motor regions which reflect the up movement and down movement, and whichever is higher, whichever average is higher, we kind of assume that the culture is moving the paddle towards that direction.
How come you have 4 of those now?
So we tried different layouts. So one was just up here, down there, and this worked better. Honestly, this, yeah.
Yeah, with respect to how we choose this layout, what was the metric to decide?
So obviously, what is the metric? So the game
decide where to put the control and.
Like the sensory and motor regions, we basically wanted to choose 2 separated kind of sub-regions of the entire chip. That's how we did that. But then in terms of what is up, what is down, obviously we started with the simpler one, more intuitive one. All of these up, all of these down, and then we measured the game performance. I'm going to tell you about the metrics that we have, and this was when they performed best. And then also, I told you this is going to be a closed loop system, so we need feedback, which is, I think, the most exciting part of the work. So how we apply feedback is that if the culture hits the ball accurately. This is when for 100 milliseconds, we give a predictable stimulus to all of the 8 stimulus electrodes with 100 hertz and 75 millivolts. But then if they miss the ball for 4 seconds, we give random stimulus. So this is a signal of 150 millivolt 5 hertz, but at random sites and for random time scales for 4 seconds. The entire duration is 4 seconds, but it randomly changes between these 8 electrodes and it stays on each electrode for a random duration of time. So basically, this is how we kind of punish them. And this is just a visualization of the system. This is an actual gameplay session. I don't know if you see anything.
Let's just see the first go.
Yeah.
I see the ball, but nothing else, right?
So we should see somehow the activity of the neurons and sensors. I think it's the lighting from the outside.
Yeah, it's several.
Yeah.
What do you mean exactly?
Okay. Predictable is every time they hit all of the electrodes at the same time for a certain duration, get the exact same input. So it is repeated every time they hit.
Highly timed across the entire.
Yes, exactly. But random is a random time duration. Yeah. But I think this. Yeah. Basically, the visualization, it's also available online, you can check it out. It shows the gameplay and the activities in both motor regions, how it's fluctuating, and then we also see the sensory region, and this is the corresponding gameplay environment that is actually being simulated at the same time. So this is all online and happening at the same time. Well, you can check this out later as well. Okay. Now, after like implementing all of this, we want to kind of measure how well we've done. We do this in kind of 2 big sections. One is just to look at the gain performance metrics. And this is like the first thing we did. We have, first of all, we have 3 types of controls. This is just a media only chip. The second one is an in silico computational model, which has random noise. The third one is REST, or basically spontaneous activity of the cultures, no stimulus. And then we have the mouse cortical cultures and human cortical cultures. And as we move in time on the x-axis, you can see that in the mouse and human cell cultures, for example, compared to media only, we get a higher percentage of higher hit numbers. So as we move up, the hits per minute is increasing, and we have a higher percentage of those in human and mouse cell cultures.
So I don't understand. So it's this time on the x-axis,
Yes.
right? And it's supposed to get go from blue to green to red. It's learning. It's learning how to play, right?
Yes. Yeah. So see, these are hits per minute. So as we go up, this means how many, like, what was the percentage of, for example, having 3 hits per minute? So the percentage of having higher number of hits per minute is increasing in the top right corner. We have other plots like line plots, which may be more clear in just the next slide. So if you still have questions, please let me know and I'll come back.
Understands the rows there. At least back.
The rows. So this says we have a lot of rallies or episodes of game drum. Whenever a ball is missed, a new rally starts. So in this entire 20-minute recordings, we have measured, we have an average of 70 episodes in all of the cultures. So in each episode, you have a certain number of accurate hits before you miss the ball. So we say, what is the percentage of the rallies that only had 0 hits, 1 hit, 2 hits, blah, blah, in each minute of the recording?
You can see, for example, for more than 5 plus hits for the human cell cultures, it's about 10%. Yeah. While in the CTO, it's about, I don't know, 1%.
0 should be 0, yeah.
Yeah. But there's many times when it doesn't even get the ball at all, right?
We do have sessions.
50%, yeah.
Still we do have sessions like that. So continuing with the game performance metrics, this is what we call average rally length. So basically very similar to what we had before, but the average number of accurate hits among all the episodes that the culture has played. And what we do is that we measure it for all of the cultures in the first 5 minutes and also the second 15 minutes. And we expect to see an improvement. And we only see a significant improvement in mouse cell cortical cultures and human cortical cultures. And the highest improvement is actually in the human cells, but everything else obviously doesn't show any significant change.
What type of cells are the controlled in breast cells?
Control is media only, no cell. So just recording. REST is the same culture, but no stimulus. So it's just a spontaneous activity, but still because you are recording from the motor regions, right? So you can compare and you can say, okay, the hypothetical pattern is moving up or down. We know where the ball is and things like that. But obviously doesn't mean to have any source
control not 0.
Sorry?
House control not 0, you don't have cells.
It's still the ball randomly.
Yes, because the game environment is still like random. Yes.
So for the neurocortical cells and the mouse cortical cells, did you perform repetitive debuggs on the same set of cells, or did you culture them again and sort of different groups of cells?
So we have like obviously more than one culture of cells, but in many of them we also do repeated recordings.
This is the average of all?
Yeah, the average of all of them. Yeah.
And I should also mention that the recordings here are just 20 minutes, and the 20 minutes which is chosen is that we don't have ability to have more than 20 minutes. This is our capability, because after 20 minutes, the neurons would die.
What happens?
Would die, the neurons.
We are
Apoptosis.
guessing temperature. Yeah.
Sorry, is that example of something going on?
This is our hypothesis, but actually we were talking to Andreas yesterday, he also had a like interesting suggestion that he would definitely bring back and test that. He said, if this duration is different on different chips like Maxwell or and MCS, maybe the toxicity comes from the actual chip, and after a certain duration of time, some chemical reaction happens, not
Between
in the chip itself, the composition of that. And that's what kind of causes the toxicity, not just the temperature of the culture. So this is
the temperature of the culture you get up.
I mean, we do our best, but after 20 minutes, a lot of them still.
How do you control it? What do you do?
It's so they are in this incubator when we are recording from them, and we could try to control the temperature.
Something like Peltier element submerged into the medium or something like that.
I think so. Yeah.
Does the intensity of the simulation somehow correlate with the longevity of the neurons?
Intensity, you mean the amplitude of the signals, for example?
Yeah, for instance, the mean rate, the simulation that you provide, once it gets correct or wrong, if you vary that, does it vary?
It would affect, but we've tried to be as efficient as possible, obviously, like as efficient as possible while still getting a response, like getting good outcomes. Okay, I don't think we have much time, so I should be faster. So we have this percentage of long rallies. Long rallies we define as the number of rallies which have more than 3 consecutive accurate hits. So again, we want this to increase in time. We compare the first 5 minutes and the second 50, like the rest of the time, and we see a significant improvement in mouse and human, but not the other groups. And also the percentage of ACEs. So this is, I mean, it's a bit misleading. ACE is not a good thing here. ACE is when the paddle misses the ball after the first throw. So we want it to decrease in time. And again, you see the mouse and human cultures show this significant decrease compared to the other which does. And then what we did, we took a further step, and because the type of feedback that we designed was just, it was not a given, right? We kind of thought about it, used some theories such as energy principle and all that, and we thought of designing this type of feedback, which I told you about, and we call this stimulus feedback, but we also implemented basically two other feedbacks we call. The first one is called silence. That's when they accurately hit the ball, the game just continues. We don't tell them anything, so the ball is hit, it bounces back and such things. But when they miss the ball, it's dead silence for 4 seconds. So instead of that random stimulus, we give them nothing.
Like punish them.
Yeah, punish them with silence instead of random, just crazy signals.
It's like kids.
Yeah. And then also, just to compare, we have this no feedback or open loop as well, which we give them no feedback. Just the game restarts, either they hit, they miss, it just goes on and there's no feedback. This is different than rest, because in rest, we have no stimulus. Here, they still give the information about the ball, but no feedback. And we compared these different feedback times in terms of all of the metrics I told you about. I just brought one of them here. We don't have that much time. And this shows that still the first type of feedback we implemented, the stimulus feedback, this is the one that shows the best performance, the highest improvement. Yeah, this is the random one. The silent is still doing a bit better than no feedback, but doesn't really show a significant improvement. Yes.
What was some of the intuition behind the random stimulus being negative?
Yeah, so you could think about it like the cultures wanting to minimize the surprise or not liking surprise, like the free energy principle. That's kind of what our ideas were based on. They don't like randomness, and they want to just get rid of that as soon as possible. Yeah. Then I told you these were game performance related metrics, but obviously we also wanted to look at activity related metrics and what is actually happening in the cultures during gameplay compared to rest. So one thing we looked at was the average cross correlation between the sensory and each of the motor regions. And we see that in gameplay compared to rest, this is significantly higher in both of the regions, so they become more correlated. Then we have the percentage of exclusive motor region activity. This means the percentage of time in each second when one of the motor regions is significantly active above noise level, but the other isn't. So they are exclusively active, not together. And this is kind of coherent with their, adaptive learning and trying to play the game. And this exclusive activity is significantly higher during gameplay compared to rest. We also wanted to somewhat look at plasticity. We chose functional plasticity and a metric that also includes spatial information to quantify the network plasticity. And what we did is that we calculated the center of activity during the entire rest state recording as a baseline. And then for each 5-minute interval of gameplay, we also calculated the center of activity, and then we calculated the distance between each 5 minutes of gameplay and then the entire rest session. And we see that, well, it rapidly changes and the plasticity is much higher during gameplay. And finally, because I'm gonna let Moein talk as well, mean information entropy. What we thought to do is to measure like the information entropy in the cultures, and we wanted to compare basically pre-feedback and post-feedback durations. So post-feedback is every 4 seconds after a negative feedback, a random feedback is applied. So we get all those 4-second intervals, calculate the binary information entropy and average out. And then prior to feedback is everything else, obviously excluding the exact feedback durations and so on. And we see that in gameplay, post-feedback has a significant increase in entropy because we introduced this randomness, but this doesn't happen in rest. This is just to validate. We thought that if the baseline sparking activity of the sensory region is for some reason higher, that would affect the information entropy. So we just normalized by the spiking activity, by the number of spikes. So the increasing relationship still holds. But in the normalization, normalized part, we see that only the prior feedback gameplay are lower than rest, and post-feedback are actually have entropies which are very close to rest. We repeated for the different feedback types. We still see, interestingly, this was one of the questions that we weren't able to fully answer in the paper. Even with the silent feedback, the 4 seconds after the silence, we see an increase in information entropy. So this was quite interesting.
After, like you interpret, like your possible interpretation is they're silenced. They're not silenced. You're just not giving an input.
Yeah. No external stimulus. Yeah, but they're not silent. We are just silent for 4 seconds.
During that silent time, are they still forming connections, or?
Sorry?
During that silent time, are they still forming connections, or no, not all?
Yeah. I mean, it's just we don't stimulate or give any boost. That's okay. And I guess I'm gonna skip this one, to be honest. So Moein can talk about our ongoing projects. If there is time, I'll come back to this. We kind of took another perspective to look at these gameplays and study the theory of criticality. I don't know if you're familiar with it, and compare between gameplay and rest. We found that gameplay is much closer to a critical state compared to rest, but that's the whole paper that is coming out in NatureCom. Sorry, I think we're going to go at 6 p.m. from Melbourne, so we need to be on time. Yeah. So I'm gonna skip this.
I wanted to talk about for 2 recent projects, but I think you can probably just one of them. Okay, so our aim was to do a direct comparison between the state-of-the-art deep reinforcement learning algorithms and our neural cultures that Forough just introduced you in the same game environment upon, but clearly during the gameplay sessions and not the rest. Okay, so recently, as you know, the RL algorithms that use deep learning and neural networks in their structures have been developed to beat human experts in various games, such as Atari games. However, these algorithms suffer from different issues like sample inefficiency or modeling the reward structure or requiring high levels of computing power and so on. And these issues suggest that these algorithms might be fundamentally different from the human learning mechanisms and be too inefficient to be a plausible model for human learning. That's why we were interested to do this study and research. So we used three deep RL algorithms in our work: DQN, A2C, and PPO. And these algorithms benefit from the deep learning advantages in feature extraction, and also specifically using convolutional neural networks in their structure. They're very robust tools in reinforcement tasks, and particularly in the games where the input is an image. We also take a step further to take into account the potential adversaries of the image input to the RL algorithms, or so-called curse of dimensionality in machine learning term. And apart from the image input, we designed two additional input information. The first one is paddle and ball position design, where we obtained 4-dimensional vectors encoding the X and Y coordinates of the ball, as well as the Y coordinates of the top and bottom of the paddle. And the second design is the ball position, which we initially divide the y-axis of the gameplay into 8 equal segments, each mimicking one of the stimulus electrodes that place code the information about the ball's y-axis position as an integer in the 1 and 8 interval, as well as the x-axis of the ball as a second element of the vector, which is similar to the rate-coded component of the stimulation in the biological culture. This design, the ball position design, is the most similar one to our biological cultures. I should also mention that, as we told you before, all of our recording sessions are 20 minutes, which is equal to an average number of 70 episodes. So in our RL algorithms, we first train them for 70 episodes and then remap them to an equivalent real time of 20 minutes. So without going into much detail, I will briefly represent the comparison results between the performance of the RL algorithms and our theoretical cultures for these three designs. And first for the image one. We examined 3 metrics: the average hits per rally, the number of aces, and the number of long rallies. And as we can see, for example, for the average hits per rally, the human cell cultures and mouse cell cultures, the blue and orange lines, reach the highest levels of these metrics. And for the aces, for example, again in the biological cultures, can see which is the lowest level. And for the long rallies of you can see, for example, the DQN which is the green one, the green line, is outperformed by all of the opponents, while the biological cultures reach the highest levels. And in the bottom row, we evaluated the improvement of the same metrics over time by calculating in the first 5 minutes in the green, shown in the green, and the last 15 minutes, which are shown in the orange. And we can see that the biological cultures, mouse cells cultures and human cell cultures, again, show significant improvements over time for all of the metrics.
But not the rest of the group.
How do you feedback
exactly what do you feed back to the Dishbrain, for example? It is exactly the. I should look at the code, but in Dishbrain we know exactly what optimization we are using, and we know what the reward and cost function. We define the cost function, and we know how to punish them if they're usable. But I will check them. We can discuss it later. Okay, so we repeated the same analysis for the paddle and ball position, which revealed the fact that the performance of the RL algorithms do not suffer from the curse of dimensionality, and all the previous results still hold. And we can see that all the results probably were similar to the previous ones. And finally, for the ball position design, which is the most similar one to our biological cultures, again, we see similar comparisons and observations. Finally, we directly compute the relative improvement in the average run lengths for the all groups, and we can see that for all of the designs, the human cell cultures show the highest improvement over time, usually followed by the mouse cell cultures. And we could conclude that the reinforcement learning algorithms show the lowest sample efficiency, having the lowest improvement over time. And this was despite the fact that these algorithms receive higher amount of information compared to the biological cultures. We also take into account the potential disadvantages of curse of dimensionality by designing two alternative input information, which show no significant improvement in the overall outcome of our studies. Okay, so you want to talk about the probability, or I'm not sure. Yeah. Okay, so do we have any questions? Okay, so in one of our ongoing projects, we wanted to compare the gameplay and rest sessions and see how the connectivity between the cultures, between the channels differs over time, and whether the evolution of these connectivities can capture the learning that seems to happen during learning. First, our challenge was we have for every recording and for each channel, we have a very long and very large time series of a spiking activity. So to make these spiking patterns more interpretable and easier to work with, we decided to use dimensionality reduction algorithms and bring each time series of the culture of each channel to a lower dimensional space. We use and compare different algorithms, including TSNE and ISOMAP. And as we can see here, interestingly, even for two-dimensional space, the first half and the second half of the recordings are easily distinguished in the gameplay sessions, but not in the recording, in the rest sessions. These figures are 4 sample cultures, and the colors represent all the channels for each half of the recordings. So, because of these differences in the representations of the gameplay and rest sessions, we were motivated to continue our study in the lower dimensional space. Also having 10,024 channels still meant having high computational complexity in our problem. So we hypothesized that most probably a lot of our channels are highly correlated, and we may be facing redundancy of information when we look at all the channels in our study. To capture and build the networks, the functional connectivity networks, and compare the gameplay and rest sessions, we designed this pipeline. So initially, we used TSNE algorithms to map all the recordings to lower dimensional space, here 3-dimensional space, and to get a smaller set of channels as a good representative of all the recording among all the recordings. We stack up all the lower dimensional representations of the gameplay sessions, and then use Tucker decomposition followed by K-memories clustering. We clustered all the recording sessions in the lower dimensional space and get the centers or centroids of the clusters as the representative channels of all the recordings. So to build the network, you use these extracted channels as the nodes of the network, and then the Pearson correlation of the spiking activity as the edge weight between any pair of nodes or channels. And very briefly, here are just a few samples of our findings. We look at the changes of the correlation between any pair of the channels from the first 2 minutes to the last 2 minutes of the recordings. And here we can see that the extracted channels from the sensory regions are shown with the orange circles, and the ones from the motor channels are the green squares. And the color of the edges indicates the increasing or decreasing of the correlation between nodes in red and blue respectively. And the thickness of the edges are related to the magnitude of these differences. And as we can see, for example, here, simply in the gameplay, we can see many more positive edge, which means the increasing correlation during time.
Sorry, I should just add, this is the average among all of the recordings, because we have the same representatives now, same 30 channels. This is averaged out between all of the recordings.
Yeah, for the gameplay sessions, we have 300 recordings, and for the rest sessions, we have 150, and we get the average network from all of these recordings shown this graph. And finally, to give you some statistics about our findings. Here we have 5 measures of the network dynamics compared from the first and the last 2 minutes of the recordings in the pink and green. For the gameplay sessions in the top row and the rest sessions in the bottom row. And for example, I'm not going into all of the metrics. Yeah. For example, for the modularity index, we can see that during the gameplay session, we can see that it decreases significantly, which means the clusters or the modules in the network become more interconnected as time goes on. Or the clustering coefficient increases significantly in the gameplay, which means the neighbors of a given node become more strongly connected between themselves. or the characteristic path length, which means the average shortest path between any pair of nodes in the network, becomes significantly decreases in the gameplay sessions.
How do you evaluate the modularity? Is it like using microscopy or something?
No. There's a function that you can see. It's an attribute of the network.
Like on a graph.
On the graph. You have lots of nodes and the edges between.
Using the recording.
Yeah. Something happened.
That's the isolation kind of decreases in gameplay. The local modules become more effective.
Okay, so as I mentioned, yeah, this is an ongoing project, and we're interested to look at more comprehensive network similarity measures, or we had a chance to discuss very briefly with Gerald, and I'm happy to discuss later the OQ, any suggestions if you have.
I think Brett's will be joining now. Yeah, I've shared.
Yeah.
Okay, yeah, sorry. Acknowledgements.
Yeah.
Oh my God.
Yeah. It also thanks all the members in Cortical Labs, and also especially Brett and Dr. Han, and also the external collaborators, especially Professor Habibollahi in Monash University. Yeah, I think that was pretty much what we wanted to cover, right? Thank you.
Are we having Brett join it as well?
Like 1 hour.
I am in fact online, if that helps.
Okay, thanks.
Hi, Brett.
Okay, so I guess everyone already knows Brett is a chief officer at Cortical Labs, and some of you had some more technical questions about the biological aspects, or the whole pipeline of how we culture the neurons. So we asked Brett if he could maybe show you some things live from Melbourne, and also answer all your detailed questions. Thanks, Brett.
Hi, everyone. Not sure if you can see and or hear me, but if there's any questions, I will do my best to answer them.
Yeah.
Or if there's a specific thing you want me to show people through, let me know.
Do you want to ask your questions?
Yeah, kind of.
Maybe?
Shall we maybe like just stop the screenshot so we can,
Yeah, sure.
Yeah.
screen?
All right.
How do you see Brett?
I'll bring him up.
It's a
Yes.
Maybe you check like that.
That's good enough, yeah.
And if you'd like to see something about like the culture, then obviously it's in the lab. So,
Show because
if you have any specific things in mind.
I think like super specific question, but
Yeah, go for it. You can-
Hi, Brett, can you hear me?
Just
Shall I speak louder?
yes, please.
Okay, great. Thank you so much for joining us. We had a bunch of, I guess, biologically relevant questions to throw at you. The first one of which was raised, during the presentation. What is your best guess as to why you can't record the neurons for more than 20 minutes? Is it like the structure of the electrode? Is it the temperature? Is it toxicology? Is it something else?
Was the question, just to make sure I heard the question properly, it was why we can't record for more than 20 minutes?
Yeah, that's it.
Yeah? Okay. You can hear me okay? There's a lot of background noise in here with the lab going on.
No, it's all good.
Okay, excellent. Well, I wouldn't say it's a guess so much. We've tested and sort of established what it is. It seems almost certainly to be the case that the temperature differential from the MEA itself, the reader itself, in about an hour or so, it raises about 2 to 3 degrees with a bit of variance. And even if we do something like lower the temperature of the incubator so that the temperature that the cells sit at is still physiologically within a range, you still, that temperature differential causes increased evaporation, which then will do stuff like change the osmolarity of the media that the cells sit in. Now, cells can tolerate changes in osmolarity within a given range, but it does seem that repeated exposures to variable osmolarity over multiple days very rapidly results in a decay of cell health and death, not to mention their performance also seems to drop off rapidly. We have, however, tested them up to 18 hours a day in our perfusion circuit. And since I have the benefit of being in the lab now, what I can actually do is show you what those look like. So here are some of them in their boxes running. And then this is actually what it looks like. So you have, these are the chips, but they're not in the reader at the moment. We're just doing another test. But essentially you have media, you have waste, you have a gas mixer here to make sure that the air is properly, the gases are properly mixed in, CO2, oxygen. We can use nitrogen if we want to control the oxygen level specifically. And then a dialysis component to actually filter out waste. And then you can, this is temperature as well, can be controlled. This is actually a heating pad, as well as where the electrode array will sit. So we can overcome this issue and it's just a matter now of scaling these up and improving the reliability of the system as well has been the focus of ours now for the last 6 months, because we got it working quite a while ago, but the hardware was unreliable, so we've been improving that. So, does that answer your question?
Yes, thank you very much.
Okay, cool. I think Gerald also has a question about this.
Couldn't you solve the evaporation just by having a closed fluidic system or covering with mineral oil?
I'm sorry, I didn't hear what that was.
Oh, couldn't you just solve the evaporation problem just by covering with mineral oil or have a closed fluidic system?
We tried that. There was some work at a Steve Potter's lab a while ago that they used this kind of like looked like cling wrap, but yeah, this material that did in theory reduce evaporation. We weren't able to replicate the results that they found, and we found that it tended to result in worse cell health over about a period of a week, and it was harder to handle. So it didn't seem to give us the same outcome that we'd hoped for. We did try it though.
No, but covering with mineral oil covering completely. Because that's standard works for in vitro fertilization, cultures and things like that. And also for, this is what people do long-term culture for in vitro fertilization, the embryos that you culture, that's basically what you normally do. Or a closed fluidic system.
Well, a closed fluidic system is what I was just showing you. Sorry, again, it's very hard to hear with the sound. But if I've understood your questions correctly, the closed fluidic system is what I was just showing you. In terms of if you're just talking about covering with some sort of membrane that prevents like evaporation from leaving the dish itself, we didn't find that was very successful.
I can, yeah, it doesn't hear. Maybe closer to that.
Hey, Brett, another question. Why?
That's so much clearer. Thank you.
Sorry?
That's
clear. Oh, okay.
That's much clearer. That's excellent. Thank you.
So you can record from 1,000 electrodes, right? Why and stimulate? Why for the Pong only kind of 12 are used?
Well, so we can record in the Maxwell system from 1024 electrodes. We can only stimulate theoretically up to 32 electrodes with the Maxwell system. However, the real number of discrete units you can stimulate is actually much lower in practice. Essentially, the way the chip works, I can't go too much into this because there's some NDAs, but essentially there's stim units around the outside that get routed to individual electrodes. But the algorithm that the hardware software uses to actually choose those, we don't have a degree of control over. And so to get actually discrete control of individual units was difficult, to put it mildly. And so we found that unless we wanted to go all the way around the outside and thereby give up the topographic encoding of information that we had previously decided to stimulate with, we were limited to about 8 to 12 electrodes, depending on the layout. And we found that 8 was the easiest to work with and encode the data, so we explored it and found good results. That doesn't mean that there aren't other ways to explore it, or that even what we've done is the best way. I'm nearly certain what we've done is not the best way. It's just something we did that worked, and we've decided to share the results. What we are doing, though, that I'll mention is going forward, is actually we're going to a system that is far less, well, we're continuing with some of the high density stuff, but we're also focusing our efforts on a system that's less dense, in this case, only 60 electrodes, but you can have symmetrical read and write. So the read density is going to drop significantly, but our control over the write, in other words, information in, will increase significantly. And we think that that's a worthwhile trade-off to explore next.
Would it be possible to read out more just to understand what the network is doing? Like maybe not during the game, but like for stopping the session and then recording for a couple of seconds, for instance, just to figure out the approximately like what's the internals of the culture is doing.
It certainly is, especially with the Maxwell system. But it's a tricky question, and it comes down to experimental design. So, I mean, we're very much at the case, not just us, but anyone in the space, of the work actually being so early and so much being unknown that there's so many experiments still to do. And that's certainly a very good idea of something someone should explore.
Thank you.
No, thanks.
We can ask about the optogenetics, like optical readouts. And we can go.
We actually spoke about that, but maybe I'll ask again, using optics for readout and write stimulation and readout.
Yeah, absolutely. And it's another project we have going in the background. And one of the benefits of our new chips, let me see quickly if I can find any of them, but they are transparent. I don't know where, there's some in the box over here, but yeah, they are transparent, the new chips that we're working with. They're all sealed. Sorry, I won't take it out just now. But yeah, they are transparent. So we could use an optical system. We are exploring a new tool to use that, but because of we're still probably quite a bit off before we put it into practice, but also because of the novelty, I'm not able to share the details on it right now. But optical out is a very, very interesting proposition, even though there are some technical challenges to implementing it, not even just to mention the density of readouts you need over time and have it read in real time. As far as I'm aware, the software to do that has not been written yet, so it would be something we'd have to write from scratch and would be, again, non-trivial, but something we're working on.
Could it be fixed the problem of temperature and we could continue our recording more than 20 minutes? Do you think this method helps us to do that?
Yeah, no, well, as I said, absolutely, we've done it. So we've done it for like 18 hours a day for 2 weeks straight and maintained cell viability at a acceptable level. In those initial pilots, there was some drop over the weeks, but the timescales of that drop is magnitudes different compared to the amount of recording compared to the previous system. So it's just a matter now of scaling up so we can get enough data to make some serious conclusions, because at the moment it's been just a couple of trials which are promising, but we don't like to sort of draw any strong conclusions until we have the robust data we want.
Any other questions, guys? I guess that's okay. Yeah. Thanks, Fred. I guess that was the most of the questions that students have. And we had lots of discussions already with them, so we have lots to bring back. Hopefully, see you soon. Can you hear us, Fred?
When you look at me, I can, or when you look at the camera, I can.
Oh. Which way to?
Yeah, thank
I think you should look at here.
Okay.
Okay, so.
Yeah, I was just thanking you. Thanks a lot. I think that was most of the questions, and we have lots of ongoing
Excellent.
discussions here. So yeah, we'll
Thank
bring
you
back
for-
a lot.
yeah.
Thanks.
Thank you very
Excellent.
much.
Well, I hope everyone enjoys the workshop and happy to answer any more questions over email. Bye, everyone.
Thanks, Brad.
Bye.
Bye.
All good. And thank you so much for organizing that.
Thanks for having us. Thank you.
Yeah.
It's been great.